with topic detection and significance of extracellular neutrophil traps in prostate cancer progression. Welcome.
Okay, so hello everyone, my name is Alexandra and today I would like to tell you about my master project, and the title is Detection and Clinical Significance of Neutrophil Extracellular Traps in Prostate Cancer Progression. My supervisor is Dr. Natalia Bednarz-Knoll from a Laboratory of Translational Oncology. So as we all know, prostate cancer is one of the most frequent cancer in men. And when we look at the statistics, sadly 1 in 8 men will be diagnosed for prostate cancer during his life, and 1 in 44 will actually die because of that. When we think about the methods of diagnostic, we have a lot of different tests and procedures. However, they're not
thank you. The next presenter will be Izabela Wojtalewicz with topic accumulation of various photosynthesizer in human keratinocytes using model of atopic dermatitis.
Okay, so good morning everyone. My name is Izabela Wojtalewicz and today I'd like to present you my master thesis entitled Accumulation of Various Photosensitizers in Human Keratinocytes using Model of Atopic Dermatitis, which I am currently conducting at Laboratory of Photobiology and Molecular Diagnostics under supervision of Professor Joanna Nekonieczna. As you can see, there are visible differences in the structure between healthy and atopic skin. In atopic dermatitis, cells do not adhere tightly to each other, which may be caused by mutation in gene of filaggrin protein, which is responsible for providing the barrier function of the epidermis by binding keratin fibers. And about 30% of patients have mutation in gene of this protein, which often cause excessive penetration of the epidermis by pathogens from external environment. One example is Staphylococcus aureus, multi-drug resistant bacteria from a group of RM pathogens. And as recent reports proven, it can live intracellularly. And this is a problem because only a few antibiotics can penetrate cell membranes in such a way as to get rid of the bacteria from the inside without harming the cell. And once the survival of intracellular Staphylococcus aureus may cause bacteria to exit the cell and resume recurrent infection. Potential solution to this problem may be photodynamic method, which involves use of photosensitizer that is irradiated with appropriate wavelength and then enters excited state, then by intersystem crossing enters excited triplet state, which can react with oxygen present in cell and generate reactive oxygen species such as singlet oxygen, hydrogen peroxide, or free radicals that can constitute oxidative stress to cell and damage structures, cell components such as lipids, proteins, or nucleic acids. Main aim of my study was to analyze profile of accumulation of photosensitizers and to assess cytotoxicity and phototoxicity of photosensitizers. And to obtain results, I used two keratinocyte cell lines, one serving as control, and the second one with silence expression of filaggrin gene. And I assess accumulation by flow cytometry and fluorescence microscopy, and cytotoxicity and phototoxicity I studied using real-time cell analysis Excel regions. In my study, I used as photosensitizers, I used rose bengal, new methylene blue, porphyrin TMPYP, and gallium porphyrin GACHP23. And now I would like to present you my results. So first, I performed analysis of accumulation by flow cytometry. And what we can observe here is that photosensitizers accumulate in keratinocytes in various ways. New methylene blue accumulate best, gallium porphyrin accumulate milias high. In case of TMPYP, we can already observe some signal from untreated cells, but after the alignment with control, we can observe only small signal, and rose bengal accumulate to a very negligible extent. And similar situation we can observe in case of filaggrin mutants. Then I performed to check how these photosensitizers accumulate. I performed fluorescence microscopy, and we can already say that these results correspond to the ones from the previous analysis from flow cytometry, but this experiment is still ongoing. And to assess safety of these compounds towards eukaryotic cells, I checked the cytotoxic effect, and selected photosensitizers are not cytotoxic to control keratinocytes. What is interesting, in case of GACHP23, we can observe like this compound seemingly even improves growth and proliferation of cells in comparison to control. And then I performed same analysis on filaggrin mutants, and also these photosensitizers are not cytotoxic to keratinocytes with silence expression of filaggrin gene. And then I wanted to check how if adding light component to the system will change growth and proliferation of cells. To this experiment, based on earlier analysis, I chose GACHP23 I studied 2 concentration and 2 dose of lights, and none of the phototoxic effect is visible. Only in case of controls, we can observe a temporary retardation of growth and proliferation after using the highest concentration and dose of light, but at the end of the analysis, the curve is on control levels, so we cannot really tell about toxic effect. And then I wanted to check if this compound in studied conditions is toxic towards bacteria. So I performed photodynamic inactivation of Staphylococcus aureus with GACHP23, and we can observe that even with smaller concentration, we can see this toxic effect. After using the higher concentration, SARS survivor drops below the limit of detection, so compound is highly effective, but even without lights, in dark control, we can observe a toxic effect of this compound towards bacteria. To conclude, photosensitizers accumulate in keratinocytes in different manner. Photosensitizers are not cytotoxic to both lines of keratinocytes. Gallium porphyrin GACHP23 is not phototoxic towards keratinocytes, and GACHP23 effectively photoinactivates Staphylococcus aureus at conditions that are not toxic to keratinocytes, suggesting the possibility for clinical applications. And thank you to all that contributes in this study. Special thanks to entire laboratory team for help and support. Thank you for your attention. If you have any question, feel free to ask.
Thank you, Isabella, for very nice presentations. And do you have a question? It's time for this.
So thank you for your presentation. Can you go back to the slide with the cytotoxicity, the results of this? Yes, this one. Why did you use the smaller concentration of these pneumatin blue? Yes.
So I performed this analysis on higher concentration as well, but in pneumatin blue is compound which is known to be toxic to eukaryotic cells, and to perform other analysis at conditions that allowed me to perform statistical analysis and also to see like real effect, I had to use smaller concentration, because for other analysis I wasn't able to perform them in higher concentration. Simply, I didn't have enough cells, like
Thank you for that.
functional cells.
Thank you for the presentation. I'm not sure if I understood correctly just the very beginning. Is it like people with atopic dermatitis have higher risk of these infections because their skin is damaged, or what is the mechanism of Staphylococcus aureus infection in this?
In atopic dermatitis, patients, about 30% of patients have this mutation, and also approximately 90% of patients have skin colonized by these bacteria already because of factors that are present in this condition. And then it allowed for Staphylococcus aureus to this invasion in the first place.
Thank you.
A silly technical question. Why you have so many drops at the longer time of incubation? Because it looks to nearly every graph. Between 40 and 50 hours, you
here?
have the drop, upper one.
Yes, here also it's here and here. It's because when it's like in case of methodology, because to perform this analysis in a longer period of incubation, I have to like I have to end this incubation. And this corresponds with these peaks. It's like change of conditions to cells.
Okay.
because I have to change medium.
But why it occur only for the longer time of incubation?
It's not. It's really here is visible here and here, here and here also, but the curves are like more established at in longer times, so it's like more visible.
Okay, thank you very much.
Thank you.
Okay, thank you. Isabella? Next presenter will be Marcin Banecki with the effect of basal breast cancer cell phenotype on the expression of immune-related markers. Floor is yours.
Good morning. Today I would like to present some ongoing research from my master thesis on how does the EMT phenotype of basal breast cancer cells affect the expression of immune-related markers. Let me start with a few words about breast cancer. So it is the most common cancer in women, and we distinguish 4 main types of this disease, the most aggressive of which is a triple negative type, or TNBC. Recent research has shown that TNBC is not actually a single uniform group, but rather is comprised of 3 distinct subtypes, and we can see them on the diagram in the right-hand portion of the slide. And my master thesis deals with the basal subtype of TNBC. An important phenomenon occurring in cancer cells is the epithelial mesenchymal transition, or the EMT. This is a process through which a cancer cell loses its epithelial phenotype in favor of a more mesenchymal phenotype. Throughout this process, the cell also gains the ability to migrate, so it has always been deemed central for tumor invasiveness. However, recent research has shown that the EMT may also influence other parameters of the cells. One such study has been conducted by Don Gray and colleagues in 2017 on a mouse model of breast cancer. In this study, the team has observed that tumors comprised only of cancer cells of the mesenchymal phenotype, as we can see on the graph B here, have been resistant against immune therapy, whereas if a tumor has been comprised only of the cells of the epithelial phenotype, as is the case on the graph A, then the usage of immunotherapy has been effective in stopping tumor growth. And there are various mechanisms which may lie behind this resistance against immunotherapy. One of them may be a decreased expression of immunoproteasome subunit genes, which down the road would lead to a decrease in the antigen presentation capabilities of the cells. Another possible mechanism for this resistance is a decreased surface level of MHC class I or MHC class II molecules in the cells. And I am speaking here about MHC class II molecules too, even though they are usually connected only with the antigen presenting molecule, with the antigen presenting cells of the immune system, because cancer cells can also express these molecules, so we think it is important to study them too. Based on the findings of the study that I've presented, our hypothesis has been that the mesenchymal phenotype of basal breast cancer cells is related with an increased immune escape. To assess whether our hypothesis is correct, we have first assessed the EMT phenotype of 23 basal breast cancer cell lines based on the expression of a panel of 76 genes. And with the scoring that we have used, it is the second column here. The higher the score, the more epithelial the cells were. Later, we have chosen 4 cell lines for further study, 2 of them epithelial. These were the cell lines HCC70 and HCC1806, and 2 of them were mesenchymal. These were the CAL51 and CAR120 cell lines. And we have chosen them based on, of course, on their EMT phenotype, but also based on the expression of some select immune-related genes by the cell lines. Our study design has been that in the epithelial cells, we are trying to induce the EMT by treating them with TGF-beta, and in the mesenchymal cell lines, we're trying to inhibit the EMT in them by treating them with an EMT inhibitor. And later, we are conducting some downstream analysis, such as Western blot flow cytometry or qPCR. The qPCR study has revealed that treating the cells with TGF-beta has been effective in inducing the EMT in the cells. And we can see that by observing the statistically significant increase in the expression of the Vim gene in the case of the HCC1806 cell line, and a statistically significant increase in the expression of the PI1 gene in the case of HCC70 cell line. These genes are both markers of the mesenchymal phenotype, so this increase tells us that our model has been correct and we have managed to induce the EMT in the cell lines. Moreover, concomitant with this EMT induction, we have also observed a decrease in the expression of the immunoproteasome subunit genes in both of the cell lines statistically significant in one case in HCC 1806, and we only have a single repeat of HCC 70, but this decrease has been quite drastic, so we are hopeful it's going to be statistically significant later on. And these findings together suggest that our hypothesis might be correct. That is, together with the EMT, there is this decrease of immunoproteasome subunit expression going on, and later it may lead to resistance against immunotherapy. Moreover, we have also studied the surface level of MHC class 1 molecules and a single molecule from the MHC class 2 group in the epithelial cell lines. And in the case of the HCC 1806 cells treated with TGF-beta, we have observed a decreased fluorescence intensity of the MHC class 1 molecules that were stained. This tells us again that the surface level probably decreased after the EMT has been induced. And similarly, in the case of HCC 70 cells, we have observed a drop in the fluorescence of the stained HLA-DR molecule from the MHC class 2 group. Together, these findings again would support our hypothesis because as the cells were treated with TGF-beta and the EMT has been induced, we observed a drop in the surface level of antigen-presenting molecules. We have also attempted to inhibit the EMT in the mesenchymal cell lines that we have chosen for study. However, here we have managed to achieve no statistical significance in both of the cell lines, and as such we cannot study these cell lines any further and we cannot analyze them. For the conclusions, we have managed to successfully induce the EMT in the epithelial cell lines by treating them with TGF-beta. This is a really good finding for us because even though TGF-beta has always been considered to be the classical EMT inducer, to our best knowledge, there has never been conducted any experiment in which TGF-beta alone has been used to induce the EMT in the cell lines that we have studied here, so a really good finding. Moreover, we have observed two things. First, a decrease in the expression of immunoproteasome subunits after the cells were treated with TGF-beta, and also a decrease in the surface level of MHC class 1 and a single MHC class 2 molecules in the cell lines. These two findings together suggest that our hypothesis might be correct and the EMT may decrease the cell's antigen presentation capabilities in basal breast cancer. And that's all. Thank you.
Thank you, Marcin, for very interesting presentations and a few questions.
Please go back to your conclusion.
Mm-hmm.
Can you specify what's the difference between second and third conclusion? Because for me it seems nearly the same.
The second one deals with the expression of immunoproteasome subunits, which is one cellular process. Another deals with the surface level of MHC class 1 and MHC class 2 molecules. The overall conclusion that there may be a decrease in antigen presentation is the same for both of these points, but these are two different experiments that show up.
Okay, thank you.
Thank you so much. I have a question about how you chose the specific cell lines. Why, we're just like, yes, exactly, with mesenchymal, why you didn't choose the cell line which has like a higher score of 7.6GS?
Yes.
Is it like,
Thank you. Because we've not only been selecting them based on the EMT phenotype, but also on the expression of some select immune-related genes. And in the more mesenchymal cell lines, this expression was quite high. And for our study, we were trying to choose the mesenchymal cell lines that would have possibly a low expression of various genes related to the interactions with immune system.
Okay, thank you. Marta? And the next presenter will be Natalia Kovtun with specificity characterization of anti-HCP.
The next one?
Okay.
W liście ja byłem pierwszy.
Bardzo,
tak? Tak.
Okay.
No?
Sorry, okay. Now will be Marcel Letniowski with long-term prediction of immunoglobulin physical and chemical degradation using the Artenius. Welcome.
Okay, so hello everyone. Today I would like to tell you about my master thesis, which was the long-term predictions of immunoglobulin physical and chemical degradation using the Arrhenius equation. And my topic is strongly related with the production of the biological drugs, and to be more specific, with a preclinical development in which the formulation step occurs. And in this step, after obtaining the final product, which in this case is the antibody, the major problem is the stability of this antibody, and in this step the major goal is to determine the shelf life of this product and also to optimize this formula to prolong its stability. And the major also the problem is that the final storage temperature in which those drugs are stored is 5 degrees, and in 5 degrees all of the degradation processes has low kinetics, and those stability studies take up to years. And for now, in case of the biological drugs, there are only two accepted methods by the agencies. It is the initial studies in stress conditions to somehow check how the protein behave in such conditions and the formulation, and the final studies in 5 degrees in which we determine the final shelf life of the product. And for now, there is no predicting tools used in case of production of biologics, but in case of the small molecule drugs, such tools are accepted by the ICH agencies. So a few words about the method that I use. So I use the Arrhenius equation, which gives the linear relationship between the rate constant and the absolute temperature. And for now, it was so far consistent with the course of simple chemical reactions on small molecules, but also with aggregation of small proteins. And in first place, in case of antibodies, this equation, this linear relationship is disrupted. So in the first place, it was said that it cannot be used for the prediction of the rate constant. But in a recent article in 2022, the scientists proposed the hypothesis that maybe this aggregation process consists of two separate pathways, which is the low temperature and high temperature aggregation, and this pattern on the Arrhenius plot consists of those two overlapping pathways. So in my master thesis, I wanted somehow to utilize this fact that this Arrhenius behavior can be observed in the low temperatures, and to try to predict the rate constant at 5 degrees. So the aim of the studies was to obtain the accurate long-term predictions based on short-term stability data in accelerated conditions and stress conditions. And the hypothesis is that this linear relationship that I was previously describing with right adjustment of temperatures maintains in case of aggregation, which is the physical degradation, and the oxidation, which is the chemical degradation of the antibodies. So as for the materials, so I use the ICH standard condition studies. So I got stability studies from accelerated conditions, stress conditions, and also the data from long-term studies in 5 degrees, which took up to 4 years. And I got 10 batches of this product. And the brief protocol, how it was done, how those calculations were done. So first I took the data from 25 degrees and 40 degrees and transformed it. And then after a few calculations, I got the rate constant in 25 degrees and the rate constant in 40 degrees. And having those two rate constants and using the Arrhenius equation, I got my prediction of rate constant in 5 degrees. And the 5 degrees studies were only used for the verification of the developed model. So as for the results of aggregation, there were 4 approaches in which I was calculating this model. The first approach was that I was taking all of the data from 10 batches and creating one prediction based on those batches, and then checking and verifying with each batch. So here are the results. So we can see that my predictions were almost remains in the confidence interval of the experimental data. So, but still we have little underestimation. The second approach was that I was creating predictions for each batch and then creating a mean value without the outliers, and then again checking with all of the batches. So here the underestimation is greater than in previous approach. The third approach was that the model was that I was creating so-called worst case scenario, in which I was taking the worst prediction, in this case the highest value of the rate constant, and then checking if this approach is better than the other 2 before. So in this case I got the overestimation of the increase of dimers. And the last approach was that I was not creating the unified model. I was calculating the prediction from one batch and then checking only for this one batch. And this, in this case also, I got the results that are a bit underestimated. And also we did the same calculations for the oxidation. But in case of oxidations, no matter what approach I used, all of the results are very much underestimated. And also to show that, I created a slope comparison of those regression lines, because they start from 0, so it is the only variable in those functions. So we can see that the aggregation slopes oscillates around 0, which means that they are somehow accurate. But in case of oxidation, there is a huge underestimation. And for the conclusions, so the developed model seems to be able to give good predictions in case of aggregation, but it needs further optimizing this model, for example, by adding the additional temperature to the Arrhenius equation to optimize it. But in case of oxidation, the predictions did not match the experimental data. This is most likely of the high variability of the analytical method used. But also it can be that the kinetics of the oxidation do not resemble the Arrhenius-like behavior, or maybe this temperature, or maybe the temperature is too high, and this is why the predictions are not accurate. So thank you for your attention.
Thank you, Marcel. Presentation.
Thank you for the nice talk. I would like to ask, how actually was the level of oxidation or this dimers formation measured?
It was measured by reverse phase chromatography, and it was the values were in reverse. You use the chromatography, yeah. And I got values from different time points.
For both.
Yeah, for both, yeah.
Hi, thank you for your presentation. And I have a question. Does the Arrhenius equation was the first idea for this prediction? Something else?
No, to be honest, no. The first idea when I started to do this master thesis was to use the AI, because the artificial intelligence, because there is a lot of work in this direction. But the problem with the artificial intelligence and machine learning and so on is that you need a lot of input data to somehow teach the algorithm how the protein behave. And the problem in my studies was that I was limited with data, and I couldn't somehow teach the algorithm how it would be, how it would behave. So I had to find other solutions, and then I came across with this paper from 2020 with Arrhenius.
Okay, thank you.
Thank you.
Thank you. And next presenter. Next presenter will be Natalia Kovtun with specificity characterization of anti-HCP antibodies used in host cell proteins, impurity analysis of chosen biological drugs, application of mass spectrometry and proteomic method.
Hello. Thank you for the introduction. I think my master diploma thesis is quite long, so I allow myself to not repeat it again. Just I need to mention that my thesis was conducted with the cooperation of Polpharma Biologics, a company that main focus is on develop the biosimilar drugs. And one can define biosimilars as somehow analogically to what generic are in comparison to the classical low molecule drugs. However, the biopharmaceuticals has the main active substance that is much larger and more complex than classical low molecule drugs. And the main active substance is usually the protein. For example, monoclonal antibodies that are produced by the living organisms. And because of that, the production of biological drugs requires many regulation, and the biological drugs need to undergo many clinical trials to prove their safety and efficiency. But in case of biosimilars, that the main substance remain the same as in their originators, they mostly need to prove that they are significantly similar to the originators. And the differences between the biosimilars are the originators usually came from the differences in the manufacturing production processes. And as I mentioned before, the biopharmaceuticals are produced by the living cells. And every living cell to maintain proper functionality requires to synthesize many different proteins that allows them to adapt to the living conditions. And living conditions for the cell lines that are used to produce different biosimilar and biological drugs are strictly defined. And once the proteins that are synthesized by the host cell lines ends up in the final drug product, they can potentially affect its stability and other properties, and therefore be dangerous for the patients. This is why it is necessary to monitor the level of host cell proteins during and after drug production processes. And for this, the golden standard method is classical ELISA assay that use specific anti-host cell proteins polyclonal antibodies that are obtained from the immunization of different animals with the cell culture material that contains the proteins that are synthesized by the cells during the drug production process. And of course, every ELISA assay has its certain limitation. And with all this said, I would like to move to the aim of my thesis, which was the usage of the mass spectrometry techniques to characterize the anti-host cell proteins antibodies specificity of the antibodies that are used in Polpharma Biologics during the biosimilar production processes. And here is a quick look of the overall workflow of my research. And now I would like to focus more on the specific method. First, I immobilize the process-specific anti-host cell proteins antibodies, as well as non-specific IgG1 molecules of the vedolizumab, which is a monoclonal antibody not specific for any of the host cell proteins, but is the antibody that is one of the drug produced in the polypharma. And to determine which of this immobilized on the microcolumns antibodies are able to bond which of the proteins present in the analyzed host cell proteins material, I used so-called bottom-up approach of the mass spectrometry technique that allows to high throughput quantitative analysis of even very complex material from the different samples. But to obtain best quality data, it requires proper sample preparation and proper sample analysis with approach that of the I'm sorry. Which requires a proper approach to analyze of the data acquisition. And this is why my first experiment was to choose the best parameters of the sample preparation and sample and mass spectrometry analysis that would allow me to obtain the best quality mass spectra that then would be converted to the highest number of proteins identified with the non-purified host cell proteins material. And as you can see here, no matter which parameter I tested, there was no significant difference between the number of identified proteins or even between the number of the peptides that were then and this is why I choose the method of the sample preparation and sample analysis based on the need to simplify and the reducing of the cost of the procedure. And this is what parameters were chosen. And with this method established, I tried to purify the host cell proteins material of the prepared columns. I tried about 15 different approach of combination of the purification condition with different elution buffers. And as you can see, no matter what I tried, the effectiveness of the purification remained on the quite similar level. And for example, here are the numbers of proteins identified within the purified material with different antibodies that was purified under different conditions with the use of the commercial buffer dedicated for the microcolumns that I used. And on this Venn diagrams, you can see how much of the identified proteins were present within the material that was purified with different type of the antibodies. And for the comparison, here are the numbers of the identified proteins are their corresponding peptide ions that were identified with the material that were purified under the most effective purification condition with the use of the acetic acid as the elution buffer. And of course, here are the Venn diagrams. But how this is all conclude? As you can see, mass spectrometry is potentially a great tool for the host cell proteins monitoring. It is not yet validated method, but the potential is there. But as you can see, there are still some of the proteins that were identified only in the material that were purified with the use of specific antibodies. This is why there are some of the proteins synthesized by the cell lines that are present in the so low level that there are first need to be bound by the specific antibodies to be detected at all. And the bias between the large number of the proteins identified within the non-purified material in comparison to the purified one might be due to the problem with the effectiveness of the purification, but there are still probably some of the proteins present that were not immunogenic enough to bring the immunoresponse in the immunization animals, and therefore there were no interaction between, there were no specific antibodies used in the classical ELISA assay and the column purification that could detect these host cell proteins. And this is further proved by the fact that some of the proteins were identified only by once they were detected by the antibodies that were obtained by immunization of the specific animal type. Nevertheless, the mass spectrometry could be a great tool for the anti-host cell proteins antibodies specificity determination, but it still requires some optimization. Thank you for your attention.
Thank you, Natalia, and we have time for one question.
I would like to ask you, what were your mock conditions, like in the last slide? Because the number of proteins detected there is quite high. So what are the mock
by
conditions?
mock, I mean the non-purified material, all host cell proteins that were not put under any purification on the columns, just analyzed by the mass spectrometry technique.
Thank you. And next presenter will be Kacper Boguszewski with topic surface presentations of cold active beta-galactosidase on Bacillus subtilis spores.
So hello, my name is Kacper Boguszewski and I am conducting my master's project in the Laboratory of Molecular Bacteriology. And today I would like to present to you the topic of my master's project, which is the surface presentation of cold active beta-galactosidases on Bacillus subtilis spores. The main goal of my project is to produce recombinant spores of Bacillus subtilis, on the surface of which would be present transgenic beta-galactosidase enzymes capable of enzymatic activity in near zero temperatures. And these spores would be later used as a tool to detect defrosting in the so-called cold chain, which is the entirety of storage and also transport done in sub-zero conditions. First, I would like to talk a bit about spore surface display, which is a technique of presenting biomolecules to the surrounding environment. And the key idea behind this method is that a spore-forming microbe is modified with a sequence coding for a, as you can see, fusion protein between a naturally occurring coat protein and some protein of interest. And during sporulation, these fusion proteins are also expressed and integrated into the spore coat, so that the protein of interest is present on the very surface. And what makes this technique special is the fact that spores are naturally very resistant to adverse conditions, such as extreme temperatures and extreme pH levels, and they lend this protective characteristic to the enzymes they present on their surface. Speaking of enzymes, to create a viable defrosting detection tool, the enzyme used has to have two key properties. First of all, it has to retain most of its activity in near zero, about 1 degree temperatures, most of its activity. And the second thing is it has to have an easily detectable enzymatic activity. For this reason, beta-galactosidases were chosen because there are many commercial substrates for these enzymes which give off a colorful reaction, and the specific beta-galactosidases, or beta-gals for short, used in this project come from Arthrobacter pseudolactophilus strain F2 and also from Rachnella sp strain R3. Both of these enzymes have been previously shown to retain much of their activity in near zero or even sub-zero in case of Arthrobacter conditions. Here's a very quick overview of the workflow of my project. However, I will be touching upon all of these subjects later in my presentation, so I will skip this slide for now. And to start, the first thing that had to be done was the construction of expression vectors. And the first thing to be done in that direction was to optimize the sequences of beta-galactosidases to expression in B. subtilis. Those optimized sequences were then synthesized de novo commercially, and then they were inserted into a PHT08 plasmid, which is a very standard vector for expressing proteins in B. subtilis. Yes, so at first, these constructed vectors were transformed into E. coli, because PHT08 can also be transformed into E. coli. And the reason for this was to quickly multiply our plasmid, which was later purified, extracted, and used to successfully transform Bacillus subtilis cells. As you can see on this here gel, this is the results of colony PCR of a few colonies of Bacillus subtilis after transformation. The transformation was successful, you can see by these bands over here and over here. So we were able to obtain transformant strains for both of our expression vectors, which brings us to the current state of the project. For both obtained strains, overexpression of their respective beta-galactosidase was induced by adding IPTG. However, and this was verified by an enzymatic assay utilizing ONPG hydrolysis. However, no difference was really seen between the induced and uninduced transformants. So an SDS page was conducted to check if the problem lies with the overexpression process itself, or whether the enzymes are overexpressed but lack enzymatic activity for some reason. And as you can see for the Arthrobacter beta-gal, we did see some overexpression, but there was none such overexpression for the Rachnella enzyme. So as of now, both of the strains will be sent and commercially they will be sequenced. They will commercially sequenced to see what seems to be the problem. And once that problem is dealt with, the future things we'll have to take care of are the following. The enzymes should be overexpressed, and their activity should be tested in different temperatures, including near zero ones, to see if they are viable as a part of a frost detection tool. If they are found viable, then various recombinant spores will be produced with different variants of a fusion protein, by which I mean that our beta-galactosases will be fused to multiple different spore proteins, for example, cod B, cod C, cod Z. Also with the use of peptide linkers and lack of those linkers, we're basically trying to build some variety here. And all of these constructed spores will be then once again tested for their enzymatic activity at near zero temperatures to, by way of elimination, find the best candidate for a frost detection tool. Thank you very much. That would be all from me. And are there any questions?
Thanks, very nice talk. I have a question about overproduction, overexpression. Have you checked other conditions of IPTG, or like try to play more with temperature to adjust overexpression?
Well, for now we were only trying to see if these proteins can be expressed in Bacillus subtilis, so we're not yet in the phase of seeing how different temperatures play into this. However, both of these proteins should theoretically should have those enzymes working at also room temperature. So we're not yet in the testing of different temperatures phase of the project.
Okay, thank you. And thank you, Kasper, for a nice presentation.
Thank you very much.
The next presenter will be Małgorzata Rozkwitalska with the role of the X-ray protein in maintaining the lysogeny stage of phage KH01 infecting clinical isolates of Clostridium difficile.
Okay, so good morning everyone. My name is Małgorzata Rozkwitalska and today I want to present to you my master thesis and its results. So the title of my master thesis is the role of the XRE protein in maintaining the lysogenic state of PHICDKH01 infecting clinical isolates of Clostridium difficile. So as an introduction, I would like to answer the question, what is Clostridium difficile bacterium and why it's difficult to combat? So this bacterium was reclassified from genus Clostridium in 2016, is a gram-positive obligate anaerobic bacilli capable of producing spores, which are, due to the high resistance, the main obstacle in fighting the infection. And Clostridium difficile infection, so in short CDI, is the leading cause of healthcare-associated infectious diarrhea globally. And it can be really dangerous because during this infection we have this prolonged state of inflammation in our intestine, and that can also contribute to the colon cancer. And as a treatment, we use antibiotics like metronidazole and vancomycin. And as we know, antibiotics trigger a prolonged state of dysbiosis in our intestine, and that's why antibiotics also been established as a major risk factor for CDI. So now we are in this paradoxical situation where treatment that is given to patient also endanger him for relapse of the disease. So of course we need better solution, and that's why we put our hope in phage therapy. And to use bacteriophages as a treatment for CDI, we need to find phages that will be specific for C. difficile and that will go strictly through this lytic cycle. And the problem here is that also for known bacteriophages infecting C. difficile display a temperate nature, a temperate lifestyle. So they are in the lysogenic state. And this problem can be potentially overcome by phage engineering. So we can, for example, knock out some protein related to lysogenic control. It can be some repressor that just stays in way to enter this lytic cycle. And that's why we're looking for in our phage PHICDKH01. So this is newly discovered temperate phage from Cyprhovilidae family that was described by our team from laboratory of molecular bacteriology. And the location of prophage in host genome was determined, and also the genome of this prophage was mapped. And here you can see the fragment of this genome, and you can see that there are genes coding proteins like integrase, recombinase, antirepressors. So this confirms this temperate nature of phage. And in my master thesis, I focus on this part of genome and especially on XRE family transcription regulator. And that's because there are no members of this family that play crucial role in regulating lytic lysogenic decision. Like for example, lambda phage is a C1 and CRO repressor. So that's why we thought that maybe this particular protein can have similar role in this new phage. So the main aim of my study was to investigate the role of the XRE protein in regulation of phage PHICDKH01 development. So in my research, I started with analysis of XRE protein binding sites, and here I used knowledge from previous work that was done in our lab. So in this previous research, we did the genome scanning with program phage promoter, and we find this putative promotory sequence marked here as P1, P2, and P3. And based on location of these putative promoters, I choose fragment for EMSA analysis. So I choose fragment this marked here S2 for checking if protein X, if XRE protein can bind to this chosen fragment. So for EMSA analysis, I started with preparing XRE protein in different concentration from 0 to 200 nanograms, and then just incubated this protein with this chosen fragment of DNA. And here you can see the results. And as you can see, at the level of 50 nanograms, we observe this band shift in gel, which indicate protein binding to DNA. So after confirming that in vitro this binding occurs, I also wanted to check if this binding of X-ray protein can change the activity of these putative promoters. So I started with construction of plasmid that harbor this region with putative promoters, along with 2 genes encoding fluorescence protein. So in this way, instead of gene 17, which encodes this XRE protein, I place gene coding GFP protein, and instead of gene 19, I place this gene coding crimson protein. And then this construct was ligated with linearized PAC19 vector. So in this way, I obtained that vector, which I named PMR01. So it looks like that. This is this construct. And based on this plasmid, I constructed another one, which I named PMR02. And it just differs in this fragment. So it also contains XRE gene under control of PBAT promoter and gene RC. And then both of these plasmids were used for separate transformation into E. coli cells and obtained cultures I used in the last experiments. So here I want to monitor putative phage promoters activity. And I started with 3 study groups, which just differed in carried plasmid. And every of this group was split into 2. To one I added arabinose to induce expression of XRE gene. And then for every prepared sample, I measure culture optical density and also measure a fluorescence of GFP and crimson protein. And here are the final results. So blue columns just showing bacteria with PMR021 plasmid. So this plasmid without XR gene. And green columns are bacteria with PMR02 plasmid, so with this XR gene. And when you look at GFP signal, you can see that after induction with arabinose, so after expression of XR gene and binding of this protein, we see this decrease in signal, which can indicate partial repression of XR genes. And most important results here is for crimson signal. You can see that after induction with arabinose, so after expression in binding of this protein, we see that there is no signal, a crimson signal at all, which indicates the complete repression of promoters genes here. Okay. And to sum up this presentation, in my work I showed that XRE protein binds to this chosen fragment of the analyzed region in phage genome, and also that this analyzed region can be important for establishing and also maintenance of the lysogeny. And also the binding of XRE protein to this putative regulatory region can inhibit activity of promoter, here as P3, which in phage genome supposedly directs expression of anti-repressors genes. So, of course, this topic needs further investigation, but understanding of the mechanisms controlling lysogenic analytic decision may help us in future to fight with Clostridioides difficile infection. And this is my acknowledgements. I want to thank my supervisor and all of my team for their help. And thank you for your attention.
It's time to discussion of this topic.
Thanks for a nice talk. Sorry if I missed it. You had this graph where you showed the level of GFP in this crimson.
Yes.
Yeah, here. That you have in this blue bars. So you have, in the last column, you have higher signal in the cells that didn't have, I guess that's this crimson, yes, inserted?
Yes.
Why would that be? Yeah.
Signal. In blue bars, here is bigger.
Yeah, then in the green one. Do I understand correctly that blue ones don't have this crimson gene inserted?
They don't have XR genes, so it shouldn't. This XR doesn't is expressed. Yeah. Here, it's only they lack this XR gene.
Okay.
Here.
Okay. Now I get it. Now it's correct. Super. Thanks.
Knowing that this XR protein have this impact on this regulation of lysogenic cycle, we can now maybe, of course we need to study this topic further, but maybe we can modify this XR gene, maybe silence this gene, that when in absence of XR gene, this anti-repressor, maybe they will be expressed, and in this way the phage will not be longer in this lysogenic cycle, and maybe it will enter this lytic cycle. And in that way, we obtain this phage in lytic cycle, which we can use further in the therapy for Clostridium difficile, because we won't use them to just kill these bacteria.
Thank you.
Thank you.
And last presenter before coffee break will be Natalia Trochowska with Bacillus subtilis spores, useful in maintaining the homeostasis of oral flora.
So hello everyone, my name is Natalia Trochowska and I would like to present to you the topic of my master thesis, which is about Bacillus subtilis spores, useful in maintaining the homeostasis of oral flora. I am conducting my research in the laboratory of molecular bacteriology under the supervision of Dr. Alessandro Negri. And I would like to start by emphasizing the importance of the oral microbiota. It is a community of symbiotic, commensal, and also pathogenic microorganisms which forms a biofilm. And it plays a crucial role in maintaining the oral homeostasis and also in protecting the oral cavity. The balance of the oral flora can be disrupted, which then leads to some diseases. And one of the most common oral diseases is dental caries, which is mainly caused by carcinogenic bacteria like Streptococcus mutans. This and other acidogenic bacteria can metabolize sugars, which then leads to the production of acids and the pH drop, which then leads to the mineralization of the tooth structure. And we know that untreated caries can then turn into some other serious oral diseases like periodontitis, which highlights the importance of preventing the tooth decay. And one of the potential solutions to this problem could be arginine deaminase. It is an enzyme that was found in Streptococcus cristatus and is also widely expressed in other bacteria. It is a part of the arginine deaminase system. So arginine deaminase system is an adaptive strategy used by certain oral bacteria to thrive in oral biofilms. This metabolic pathway breaks down the arginine into citrulline and also ammonia, which raises the pH level in the mouth. This higher pH reduces the risk of the cavities and also reduces the risk of the tooth decay. But the production of this protein can be too expensive, so a more affordable and a cheaper solution can be Bacillus subtilis spores, which can display proteins on their surface. So Bacillus subtilis is a bacterium that forms spore. It undergoes the process of sporulation under unfavorable conditions. It is non-pathogenic and also is also generally recognized as safe. And here are the main structures of the Bacillus subtilis spore. Here is core, inner membrane, germ cell wall, cortex, also outer membrane. And the essential for my project is a coat. It consists of several protein layers, which provide the mechanical protection to the spore. And also what is important that the spores of Bacillus subtilis has are resistant to extreme heat, the lack of nutrients, UV radiation, drought, and also exposition to various chemicals. What is the spore surface display? It is a technique used in molecular biology. It is used to present high stability proteins on the spore surface that will retain their functionality. And it consists of 4 main steps. First, the desired gene for the desired protein is fused with the gene encoding spore coat protein, which is then induced to the Bacillus subtilis. During sporulation, this fusion protein becomes a part of the spore coat. And then this spore, which are displaying the protein on their spore surface, are then released, can be purified, and also used, for example, for the production of enzymes, oral vaccines, drugs, and more. The main goal of my project is to create and produce spores of recombinant strain of Bacillus subtilis, which will be able to display arginine, the immunase, on their surface. And here is the research plan. The first step was to create the gene fusion and transformation of the E. coli DH5 alpha, then integration of the genetic construct into Bacillus subtilis chromosome, creation of recombinant strains of Bacillus subtilis able to produce ARC-A on their spore surface, then production of those spores, and the last step is the assessment of the ability of recombinant spores to increase pH level in arginine-containing environment. And in this step, I obtained a construct with the gene fusion, the gene encoding code Z, the code protein, and also ARC-A. And then I transformed the E. coli DH5 alpha with this construct and screened obtained clones for the presence of ARC-A's gene sequence. Then to check the correctness of this construct, I performed a restriction analysis. I used an enzyme that cuts within the sequence of ARC-A. Then I compared these results with the simulation in agarose gel separation in the SNAP gene. And the empty plasmid had around 9,000 base pairs, when the expected product had around 2,700 and 7,600 base pairs, and is visible here. And then I transformed the Bacillus subtilis 168 with this construct, and then to check if the genome of Bacillus subtilis had this gene fusion, I performed a PCR using the forward primer for code Z and also reverse primer for ARC-A. And as a negative control, I used a genome of Bacillus subtilis 168, and also here's the result. The proper gene fusion had around 1,600 base pairs. Also, I produced the spores, and here are the spores produced by the wild type, and here are the spores of my recombinant strain. And there are no morphological changes between those strains. And also, we can assume that this gene fusion does not interfere with the genes that are correlated with the sporulation process. And here are the future plans. I want to check the spores for the presence of ARC-A protein using Western blot. Also to check the ability of spores to produce ammonia in the solution with arginine, to create a fusion protein of ARC-A and code B and check its activity, and also the comparison of both fusion proteins on their effects. Thank you for your attention.
Thank you, Natalia.
Thank you. So I was just wondering, because I don't think anyone would disagree with the fact that if you have pH very low in the mouth, that can lead to caries. So everyone obviously will agree with that. But with your approach, so if you want to elevate it, I just wonder how you will be able to control the pH then, because too much of a pH elevation will probably disrupt the sort of microbiome in the mouth, which can also be a problem.
Thank you for your question. I think that this will need further studies to apply the correct amount of these spores to not increase the pH too much. So I think we'll...
Those dependence, basically.
Yeah.
Okay, thank you, Natalia.
Thank you.
And now... I would like to welcome you to coffee break, and we will come back here 10 minutes to 12. after break, and now presenter will be Jakub Buniewicz with topic design, production, purification and functional analysis of C949N and R205L gain on function variants of the complement C2 protein.
Thank you, and good morning everyone. So once again, the topic of my presentation is design, production, purification and functional analysis of S94N and R205L gain of function variants of the complement C2 protein. So let's start with what is a complement system. It's part of the innate immune response against pathogens. It's composed of more than 40 proteins that are present mostly in serum. There are 3 pathways of activation called classical, lectin, and alternative. And they work in a cascade manner where one protein cleaves and activates the next protein in the cascade and so on. Complement system has 3 main effector mechanisms, which are opsonization, release of the pro-inflammatory molecules, and lysis of the target cell. And here is the summary of 3 complement pathways. As you may see, at different stages, pro-inflammatory and opsonizing molecules are released, and the end result of the cascade is lysis of the target cell. And now, here is the key player for my research, which is C2 protein. It's involved in early stages of the classical and lectin pathway activation, and it, together with protein C4, forms a complex called C3 convertase. And this complex is crucial for the next steps of the cascade. The structure of C2 is, as you may see, it's built of 2 subunits, C2A and C2B. Part C2B is composed of 3 CCP domains, which stand for complement control protein, and they are involved in ligand binding. And C2A is composed of von Willebrand factor A domain, which is also involved in ligand binding, and the serine protease domain, which is responsible for the proteolytic activity of this protein. And going back here, the second important protein for my research is factor B. Factor B plays a very similar role to C2, but in the alternative pathway. And in fact, those proteins share 39 amino acid identity, and they have identical domain organization. And mutations in factor B are often associated with complement hyperactivation, which is observed in patients with different kidney diseases, for example. And in fact, 2 gain-of-function mutations in factor B were recently described, and they are listed here. And our idea was to translate those mutations from factor B to C2. So we aligned sequences of factor B and C2 to find the corresponding sites in those proteins, and we wanted to put those amino acids here to the corresponding positions here. And what is important is that both of those mutations are located in C2B subunit, as for now every described gain-of-function C2 variant has a mutation in part C2A. So this is the first attempt to obtain mutants with mutations in C2B. So the aim of our study was to obtain these 2 variants of the C2 and to assess their activity. And our hypothesis was that these variants activate classical pathway more efficiently than the wild-type C2. Here are the methods that I used. As I already said, we first aligned sequences of C2 and factor B. Then we designed mutation-containing primers for the site-specific mutagenesis. And mutagenesis was performed on PSEP4 plasmid, which is suited for the protein overexpression. And it had wild-type C2 sequence in it, and also a histag sequence, which allows for the purification later. So after the mutagenesis, we transformed DH alpha E. coli with those plasmids, sequenced them to confirm that those mutations are there, and then we transfected ex vitro cells to produce those variants. Ex vitro cell line originates from the Chinese hamster ovary cells, and they grow in high-density suspension culture, and they allow rapid and also high-yield protein production. So after the production process, we purified our variants with the affinity chromatography using Histrap column, which binds those histidine residues from Histac. And then we analyzed our products with SDS-Phage and Western blot. Here, as you may see, the result of SDS-Phage showed that we did obtain only one of our variants called R205L. There is a band for it, and there is no band in the fourth lane, which means that it suggested that we did not obtain the second variant. And it was also confirmed with Western blot and anti-Histac antibodies. Again, we have a band for variant R205L, but not for the second variant. So we proceeded with the functional analysis of the obtained variant. And to do that, we performed an assay called convertase stability assay. It allows for the assessment of activity and stability of the classical pathway convertases at different time points. And because the end result of the complement cascade is lysis of the cell, so the activity can be measured by a percentage of lysed cells. And here are the results. We use a normal human serum because it contains complement proteins and also heat-inactivated NHS, which serves as a negative control here. And as you may see, NHS supplemented with our variant showed increased activity measured by percentage of full lysis compared to pure NHS or NHS supplemented with wild-type C2, which confirmed its gain-of-function character of this mutation. So, to sum up, we did obtain R205L variant, but not the second variant, and we successfully showed the gain-of-function character of this mutation. And now it is important because this is the first gain-of-function mutation located in part C2b and not C2a. And in general, part C2b dissociates from the C2a when the convertase is formed. So the C2a is a part of the convertase, but C2b is not, which means that this mutation has to somehow improve the binding of C2 to C4 and not the convertase activity itself. So we believe that this discovery might be the first step to design a competitive inhibitor that binds C4b better than the wild-type C2, but such inhibitor would be devoid of enzymatic activity. And it will result in the inhibition of the classical pathway before the pro-inflammatory molecules are released, which might be important in diseases where there is a chronic inflammation caused by the complement hyperactivation. And that's all from me. Thank you.
Thank you, Jakub. I have one question.
Yes.
You didn't get second variants. Why?
We believe that might be caused by the formation of secondary mRNA structure, the hairpin loop, which just inhibits, which silences the expression or the production of this protein. And by the way, this is not because our team already produced many gain-of-function variants of C2 with mutations in different spots. And it's not the first time that we see that some variants just cannot be obtained.
Okay. Thank you so much. Second question? Do you have a question to Jakub? Okay. Thank you.
Thank you.
And next presenter will be Marta Greglewska with topic expression of recombinant C2 gain of function complement protein in prokaryotic system. Welcome.
So, good morning everyone. My name is Marta, and today I would like to present you my master thesis, which I done in the laboratory of cell biology and immunology, and the title is The Expression of Recombinant C2 Gain of Function Complement Protein in a Prokaryotic System. So, complement system is an important part of our innate immunity, which helps to fight pathogens, and it also induces many inflammatory responses. C2 protein plays a crucial role at the very beginning stages of its classical pathway activation. It is cleaved by the C1 complex into the two fragments, which one C2A protein, is forming the C3 convertase, a key enzymatic complex of the whole complement. At the opposite side, in the alternative pathway, we've got another protein, factor B, which has a similar function to the C2 protein. Also, they share a high degree of the amino acid sequence identity, and based on all those facts, the research group from my department decided to translate into C2 proteins some point mutations which occur in the factor B. Those mutations occur in the patients with rare kidney diseases, and they result in the hyperactive phenotype of this protein. So as a result of this research, they obtained the C2 gain-of-function mutants, which are able to enhance the complement-dependent cytotoxicity of type first anti-CD20 monoclonal antibodies, such as, for example, rituximab. And I also would like to say that those mutants are currently designed to be produced in the eukaryotic system, which has some drawbacks, mainly the low production efficiency connected with the high costs. It is also important to say that the research group has the patent protection for this discovery, and currently there are some companies which are interested in buying this patent, but they have one request. Mainly they want to have a possibility to produce this protein in the prokaryotic system. And the whole idea with the prokaryotic system came up because in comparison to the eukaryotic system, it has many more advantages. It's a quick and cheap process which leads to the more efficient production. Also, it allows for the many applications which help to popularize the product, and also it increases its commercial attractiveness. Nevertheless, there's one major problem with producing the eukaryotic proteins in the prokaryotic system, mainly the lack of post-translational modifications, such as, for example, the glycosylation or formation of the sulfide bonds. So, taking all these informations into account, I decided that the aim of my project will be the expression of functional recombinant C2 gain-of-function complement protein in the E. coli prokaryotic system. And my hypothesis is that the expression of this protein is possible to obtain in the prokaryotic system. So here you can see the methodology. I started with a SnapGene project, and after that, by the PCR method, I obtained all the DNA fragments which were necessary later for the Gibson assembly cloning. All the plasmids which I obtained were isolated and then sequenced, and after that I transformed them into E. coli BL21 strain without long and empty proteases. Next step was performing of the overproduction by adding the IPTG and also by overnight culture with shaking. I obtained the overproduction, and the next step was the fractionation. By using the cold osmotic shock, I was able to obtain the periplasm fraction, and by using the buffer with a lysozyme, I isolate the cytosol fraction. Also, the medial and insoluble fractions were collected. Simultaneously, I would like to perform the purification on the column with the nickel beads, and all subjected fractions then performed to the SDS page electrophoresis connected with the Kumasi blue staining, and also the Western blot analysis with monoclonal antihistac antibodies. So on this slide, you can see one of my projects. I decided to choose the pierced plasmid, which has a strong T7 promoter. Also, the expression could be induced by adding the IPDG, and it also carries the ampicillin resistance gene, which is necessary for the selection. The sequence for C2 wild type and gain-of-function gene was optimized for the codon usage of the E. coli, and also they have a histac, which is necessary for the purification. In order to solve the problem with the formation of the sulfide bonds, I decided to add signal sequences. I chose 6 different ones, and the presence of those signal sequences is necessary to allow the transport of the product into the periplasm where the sulfide bonds can be formed. And here is the result of the PCR, which I managed to obtain all DNA fragments which were necessary for the cloning process. I obtained the signal sequences, pierced vectors, C2 wild type, and C2 gain-of-function DNA fragments. As a result of Gibson assembly cloning, I managed to obtain the 6 different plasmids with all desired signal sequences. Their presence was confirmed by performing the PCR when I checked the presence of specific DNA fragment which is present in this C2 protein, and also the sequence correctness was confirmed by the sequencing. After that, I performed the fractionation. I isolate 4 fractions: medium, periplasm, cytosolic, and insoluble fractions. Unfortunately, in this experiment, I did not observe any additional bands which suggest the presence of the overexpressed product. I expect the additional bands at the high approximately referring to 80 kilodaltons, which is the mass of C2 protein without glycosylation. Those results were also confirmed by the Western blot analysis, where monoclonal antihistaminic antibodies did not detect any specific products. So to sum up my presentation, I managed to obtain all of those plasmids which I designed, but the results suggest that the C2 wild type and C2 gain of functions proteins are probably not expressed in this system, or they are maybe produced, but at the very low level. And here are the possible reasons for the obtained results. First of all, the product of the expression could be toxic for the bacteria. Also, the strain which I use for the overproduction could be inappropriate in this approach. And the problem could be also with the secondary structure of mRNA. Despite not promising results, I'm still doing another experiments. Currently, I focus on the purification on the column with nickel beads to finally verify if this expression is present. And if these experiments show that there is no expression of those proteins, I would like to transform the obtained plasmids into the another E. coli strains. For example, the Rosetta, which has different codon usage for the eukaryotic proteins, or Arctic Express, which has some chaperones to help in the protein folding. Also, it may turn out that the problem is more complex and further analysis, including the mRNA structure, could be needed. And at the end, I would like some acknowledgements. I would like to thank to my whole team, especially Małgorzata Stasiłowicz, for the all help and constant support. And also, I would like to thank the Laboratory of Molecular Bacteriology for all help and received advices. And also, thank you for your attention.
Thank you, Marta, for a very nice presentation. It's time to discussion.
What was the medium? Was it LB, or did
Yes,
you try
LB.
richer medium?
LB, and for the all plasmids, it was LB with ampicillin.
So I would advise trying richer medium, like with more yeast extract, with more peptone, with stabilized pH. Try to add the phosphate buffer, glycerol for stabilization, and add aeration. It also affects expression. We often get no expression until we, let's say, shake in different flasks that allow more aeration.
Okay,
This
thank you for
also
this
may
suggestion.
help.
Have you tried to check if you have a transcript with RT-PCR from your plasmids?
Not for now, but also we're considering doing the RT-PCR because, like, for now we have this purification step, and after that we plan those things which I show on the slide. But recently we also thought about it, that the problem it will be also with the transcript.
Okay, thank you.
Thank you, Marta. And next presenter will be Leon Iwaniuszyn with topic assessment of complement receptor 1 CR1 function by detection of C4D fragment on human erythrocytes.
Okay. So, hello. My name is Leon Iwaniuszyn, and topic was, so I will present you this topic. So firstly, I would like to remind you how complement receptor works, how classical passive complement system works. After attachment of antibodies to target cell, antibodies form hexamer and allow C1 complex to attach, which cleaves C4 and C2 proteins, cleaves C4 and C2 proteins, which create classical convertases and allow further complement cascade. And finally, membrane attack complex is formed and target cell is killed. Such potent mechanism of cell cytotoxicity has to be strictly regulated, and it is regulated by soluble convertase regulators like C4b binding protein and factor H, and membrane-bound convertase regulators like membrane cofactor protein and complement receptor 1. I was using erythrocytes as model cells in my project because they lack of expression of MCP. Therefore, CR1 is the only convertase regulator present on their surface. And CR1 has several functions, but I was interested in describing of its cofactor activity, which is based on its binding to C4b molecule and creating binding site for factor I serine protease, which in turn cleaves C4b into inactive C4d and C4c. Therefore, further complement activity is impossible. Here I would like to mention that C4d is normally bound to the membrane, but some its fraction is also present in solution and may be detected using ELISA technique. Here is the domain structure of CR1 molecule, and I would like to mention here that some its short consensus domains are both specific to, are responsible for both binding C3b and C4b molecules. And in contrast with, we have reports describing inhibition of CR1 cofactor activity in C3b cleavage, but there is still no information about such inhibition of CR1 cofactor activity in C4b cleavage. And I was checking if it's possible to inhibit CR1 cofactor activity in C3b cleavage by addition of blocking antibodies, which are specific to domains which are responsible for binding C3b and C4b binding. To check it, I was isolating erythrocytes using centrifugation in a density gradient. I was using lymphoprep medium, and erythrocytes were stored in Elsevier solution. Next, I was incubating obtained erythrocytes with different combination of sera and antibodies in order to create C4b molecules. And these C4b molecules, well, their amount was analyzed using sandwich ELISA and flow cytometry. C4d was C4d found bound to the membrane of erythrocytes was stained by immunofluorescent staining, and primary antibodies were specific to short neoepitope, which is formed after proteolytical cleavage by factor I, and secondary antibodies were conjugated to Alexa 488 fluorophore. And C4d found in solution was measured by sandwich ELISA, and capture antibodies were also specific to this short neoepitope. Primary antibodies were just classical anti-C4d rabbit antibodies, and secondary antibodies were conjugated to horseradish peroxidase enzyme. Here I would like to present you a composition of probes. There were three groups of probes which differ in number of erythrocytes per sample, and each group had negative control with heat and activated serum. Therefore, no complement activity was possible here. For positive control with normal human serum and sensitizing antibodies, and samples with blocking antibodies. Also, no erythrocytes probes were made, and we were expecting the lowest value of formed C4d. Surprisingly, the signal from no erythrocytes samples were pretty high, and they are comparable with our positive control, which may be explained by presence of soluble convertase regulators like C4BP and factor H in probes. Nevertheless, negative control with heat inactivated serum had pretty low amount of newly formed C4d, and there is trend that increase in the concentration of blocking antibodies binds with decreased, correlates with decreased amount of newly formed C4d. Flow cytometry showed that erythrocytes with incubated without sensitizing antibodies in normal human serum had no C4d on their surface, but incubation with sensitizing antibodies and normal human serum gave us pretty high amount of this C4d. And we used only small concentration of of blocking antibodies here. Therefore, it didn't affect it highly. Finally, I would like to conclude that addition of high concentration of blocking antibodies binds with decreased amount of newly formed C4D on erythrocyte surface, and that the further optimization is demanded using, for example, serum-free system or sera depleted in soluble convertase regulators like C4BP and factor H. So, and thank you for your attention, and I'm going to be glad to answer your questions.
What is your future plan in these studies?
So, how I told, we need to repeat these experiments with, for example, for flow cytometry, we need repeat these experiments with higher concentrations of blocking antibodies, but we were run of blocking antibodies, therefore we had results only from low concentration, so with this peak describing a high amount of C4D on erythrocyte surface. And also we need to repeat all these experiments in a synthetic model with purified proteins or using serum depleted in convertase regulators.
Okay, thank you so much.
There you go.
Next presenter will be Fabian Trzciński with topic anti-neoplastic effect of nutraceuticals and their combination with chemotherapeutics on leukemia and breast cancer cell line.
Okay, so good afternoon everybody. My name is Fabian Trzciński and I would like to present my project entitled Anti-Neoplastic Effect of Nutraceuticals and their combinations with chemotherapeutics on leukemia and breast cancer cell lines. Starting with the basics, chemotherapy is a systemic treatment used to either kill cancer cells or limit their proliferation and metastasis. The major issue with chemotherapy is a very narrow therapeutic index of chemotherapeutics drugs, which means there is a very small dose range that can effectively treat cancer without causing serious toxic effect. Additional problem is the development of resistance by cancer cells, which brings a need to increase the dose of chemotherapeutic, which is only possible until the serious adverse effects occur. Strategy to limit these problems is chemotherapy supplementation. By using compounds not commonly recognized as chemotherapeutics, we can increase the outcome of the treatment by increasing the overall anti-cancerous effect, reversing the resistance, and lowering the toxicity. In this research, we investigated 3 compounds commonly found in daily use food products: curcumin, gainstain, and sulforaphan. All 3 of them demonstrate very wide range of mechanisms of action, which results in multiple cooperative effects. Most of the research on these compounds were done on concentrations outside of the bioavailable range, and therefore these are not applicable in vivo. In my research, I used bioavailable doses of these compounds with a prospect for future clinical appliance. The supplementation effectiveness was tested on 2 compounds. Doxorubicin, used mainly in treatment of solid tumors and induction chemotherapy of leukemias, and 6-mercaptopurine, used mainly in long-term maintenance chemotherapy of leukemia. Therapeutic doses of both compounds were used in this research. Treated cell lines represented 3 diseases that require chemotherapy as the main form of treatment. These were acute lymphoblastic leukemia, Burkitt lymphoma, and triple-negative breast cancer. The particular cell lines were chosen as the extension of previous studies. And for ALL, these were Jucrucat and MOLD4. For BL, Raja and Ramos, and for TNBC, MDA, MB 231 and 41. I would like to point out that up to this time, most of my research were done on the lines of leukemia and lymphoma. The aim of the project was to analyze the effectiveness of nutraceutical supplementation of chemotherapeutic treatment. This was meant to be achieved by analyzing the effectiveness of individual nutraceuticals, their combinations, and their combination with chemotherapeutics, as well as the assessment of the type of cellular death induced by the formulated combination. The effectiveness of compounds was analyzed by measuring the cell viability with the use of MTT assay. As we can see, sulforaphan exhibited the most potent effect and decreased the viability in all 4 cell lines. Genistein at high concentration decreased the cell viability in ALL lines, but what's interesting, at the same concentrations, it stimulated the viability of the cell lines of Burkitt lymphoma. As the sulforaphan was the most effective, we decided to combine its dose of range with fixed doses of genistein and curcumin. And as we can see here, the addition of genistein could enhance the effect of sulforaphan, and that effect was not such apparent in curcumin. Similarly, the combination of genistein, curcumin, and sulforaphan was not more effective than the combination of just sulforaphan and genistein. Therefore, we decided to exclude the curcumin from the next tests and selected doses of 0.2 of sulforaphan and single dose of genistein. As sulforaphan can be toxic to the healthy cells, we compared the selected dose to the results of the previous tests performed on the line of human fibroblasts. The selected dose should, as we can see here, the selected dose of 0.2 should not be toxic to the healthy cells. As we can see here, the supplementation with sulforaphan can increase the effectiveness of 6-mercaptopurine treatment in all 4 cell lines. The addition of genistein here can increase this effect only in the cell line of MOLD4, and in the cell lines of Burkitt lymphoma, it just interrupted. In the doxorubicin treatment, we observed a similar effect of sulforaphan with a small exception for MOLD4, where particular doses of sulforaphan could increase the viability of cells treated with doxorubicin. Here, we didn't observe increasing effect of genistein, but just as before, the genistein interrupted the effect of the treatment in the cell lines of Burkitt lymphoma. The results of initial test of flow cytometry on MOLD4 demonstrated that the addition of sulforaphan didn't increase the contribution of necrosis in the induced cellular death, in the cell death induced by the doxorubicin treatment. At the same time, it confirmed the previous effects that addition of sulforaphan can increase the viability of the MOLD4 cell line treated with doxorubicin. To conclude, sulforaphan at bioavailable concentrations demonstrates a potent antineoplastic effect on both ALLN and BL cell lines. The addition of genistein enhances the antineoplastic effects of sulforaphan on ALL cell lines, and supplementation with non-toxic dose of sulforaphan enhances the effectiveness of 6-mercaptopurine treatment on all the cell lines and the effectiveness of doxorubicin in Jurkat-Rajeh and Ramos lines. Additional supplementation with genistein can further enhance the anticancerous effects towards LLL lines, but interrupts the effect towards BL lines. And the supplementation of doxorubicin treatment with sulforaphan does not increase the contribution of necrosis in induced cellular death in the MOLD4 cell line. And at this point, I would like to say thank you to the entire team of Department of Cell Biology and Immunology, especially my supervisors, Dr. Patrycja Koszałka and Dr. Grzegorz Stasiewicz, and Professor Jacek Bigda, who provided a substantive support. Thank you for your attention.
consider its mutagenicity in the lower concentrations below the cytotoxicity threshold?
If you can repeat the last part of the question.
You found that one of the compounds you used is cytotoxic at some concentration.
Yes.
Right? Have you considered its mutagenicity in the lower concentrations below this threshold?
No, I'm afraid no.
But you want to check it because sometimes it's like cytotoxic at upfront concentration and maybe mutagenic at lower concentration.
Mm-hmm.
And the second question, the compounds you use may interact with the doxorubicin.
Yes.
So consider how these interactions, direct interactions between these compounds may affect your results, especially in the
Mode 4 lines.
Sorry?
I think in the mode 4 line, yes, because it was the increase. Yes, I thought about that, and for example, it was totally my guess, but the sulforaphan is a very potent antioxidant, and doxorubicin, one of the mechanisms of action of doxorubicin is increasing the reactive oxygen species in the treated cell lines. For I thought it might be connected, and the sulforaphan might just decrease the level of reactive oxygen species in the treated cell lines. Mm-hmm. Yes.
Maybe I have a question. Sometimes we observe that combination drugs impact less effects. Do you know why?
Combination drugs have less effect,
Yes.
have lower effect. Yes, I'm not sure here, but no. Okay, I cannot move it. But it's possible that it can be the false signal of the MTT assay, because the signal that it gives us, it's not entirely cell viability. It also depends from the metabolic activity of the cells. So the more active metabolic active cells can process more tetrazolium salt in MTT assay, and at the same time they are giving the more powerful signal. So these drugs may sometimes just increase the metabolic activity of the cells, at the same inducing the apoptosis, but giving the false signal in the MTT assay.
I see.
Mm-hmm.
Thank you so much. The next presenter will be Anna Ziółkowska with development of purification methods of virus-like particles based on capsid protein of human norovirus.
Works, yeah. Hello everyone, my name is Anna Ziółkowska.
Sorry, ladies and gentlemen. We have 5 minutes.
Okay. It's okay.
Never mind.
So my name is Anna Ziółkowska. My master thesis is development of purification method of virus-like particles based on capsid protein of human norovirus, under supervision of Professor Bogusław Szewczyk and direct supervision of Dr. Martyna Kramer-Balska. At first, I like to focus on virus-like particles, or VLPs. They are a capsid without the genetic information that consists of virus protein or proteins, different. It can be enveloped, non-enveloped, depending on the virus type. And VLPs can be used as vaccines or as vectors for mRNA delivery. And in my thesis, I focused on human norovirus, which its capsid you can see here. This is norovirus strain G24, and I used VP1 protein of this strain, the protein of the strain, which you can see here in its monomeric and dimeric form. It is worthy to mention that This VP1 protein is secreted from the host cell and self-assembles into VLPs. And also there are currently clinical trials of vaccines based on VLPs against human norovirus. My aim with this study is to produce virus-like particles based on human norovirus VP1 protein in insect and in mammalian cells, and to establish a scalable method of purification of these VLPs that is comparable to ultracentrifugation method used in small-scale purifications, and to check whether the different pH of the buffers used in ion exchange chromatography influence the assembly or disassembly of VLPs. My general workflow was obtaining VP1 protein first in insect cells, then optimization of the ion exchange chromatography method, and checking the electron microscopy pictures of the chromatography fractions, whether there's VLPs or not. Then I focused on mammalian cell system. Then I cloned plasmids, obtained the VP1 protein, did the anion exchange chromatography with already established conditions, and the electron microscopy of selected chromatography fractions. And first, baculovirus expression system. It uses baculovirus as a vector and insect cells as the host cell. And first, I established the dynamic of infection. And here you can see that the best results, the biggest yield of the VP1 protein was obtained in MOI3 and on day 5 of infection. But for my purposes, I used MOI1, which is multiplicity of infection. And here you can see the obtained protein on Kumasi stain and on Western blotting. Then I focused on anion exchange chromatography. And here I changed the parameters, which are pH. And the literature suggests that in pH 9, this VP1 protein of this particular strain should disassemble and into mono and dimeric forms of VP1 protein. So this is the optimization of the chromatography. Here the goal was to obtain the protein in 2 fractions per elution so that the protein density would be high. And as you can see in pH 7 and pH 9, unfortunately the protein was not dissociating from the resin quickly. In pH 3, there is no protein because the protein didn't bind to the resin at all. And in pH 6, I've obtained the protein in 2 fractions per elution. Next, I focused on mammalian cell transfection, and here I wanted to compare 2 plasmids, which is pcDNA 3.1 and pcDNA 3.4 topo, which are both used for mammalian cell transfection, but the pcDNA 3.4 topo is used in cells that grow in suspension. And here I have already had pcDNA 3.1 with the gene of interest cloned, so I only did pcDNA 3.4 topo. And here you can see in-situ amplification, the enzymatic digestion, and the final checking whether the cloning went well, and then it was sent to sequencing, which came back positive. So I've obtained VP1 protein in XP293 cells, which are mammalian cells. And here you can see the comparison of post-transfection medium and precipitate medium that will be put onto a column. And it seems that pcDNA 3.4 topo gave a higher yield, but you don't really see the difference in Kumasi brilliant stain. So that's why it seems. And then I did anion exchange chromatography in pH 6. And as you can see, the results are similar to pH 6 with baculovirus obtained protein, but in pcDNA 3.4 topo, unfortunately, the protein was present in more than 4 fractions total. And this is the final comparison of the fractions that had the most protein in every single chromatography. And as you can see, the purity that I've achieved is better than what I've started with. And the protein is present in every single fraction except the pH 3, where the protein didn't bind to the resin. And finally, this is the comparison of electron microscopy analysis of different pH, of chromatography done in different pH. And as you can see, in pH 3, there are no virus-like particles, and in pH 6, 7, and 9, there are virus-like particles, which is surprising because the literature suggests otherwise. And in pcDNA 3.1 and 3.4, there are also virus-like particles present. In conclusion, VP1 protein was efficiently secreted to the medium and in both insect and mammalian cells, and precipitated with PEC 6000 and sodium chloride. Mammalian cell transfection of plasmid pcDNA 3.4 topo seemed to have resulted in higher VP1 protein yield than pcDNA 3.1. Ion exchange chromatography could be an alternative to ultracentrifugation method. And transmission electron microscopy has shown the characteristic morphology of VLPs. And they were present in pH 6, 7, and 9, but not in pH 3. And this is my bibliography, and thank you very much.
Very nice. I was wondering, could you comment on the double bond pattern that you're seeing in your purified protein?
Yes, so sometimes the VP1 protein is cleaved on the, I think, C-terminal end. It is not really known why, but it sometimes happens. And also, there can be monomeric or dimeric forms of the protein, so that is probably the reason why.
Okay, thank you. Next presenter is Eva Raszewska with analysis of the effect of glycosylation inhibitors on the SARS-CoV-2 virus structural protein production.
Hello everyone, my name is Eva Raszewska, and today I would like to show you results of my master project, which title is Analysis of the Effect of Glycosylation Inhibitors on the SARS-CoV-2 virus Structural Proteins Production. My supervisor is Professor Ewelina Król, my lab supervisor is her PhD student Ewa Wiśniewska, and all my work I did in laboratory of recombinant vaccines. So, SARS-CoV-2 virus, I think it's well-known virus for all of us. It's a single-strand RNA virus which belongs to the family Coronaviridae. Here you can see that this virus has 4 structural proteins which were very important during my laboratory work. This virus can cause disease called COVID-19, and here you can see how many confirmed cases and deaths it caused until this day. So I think that's quite a big issue, and such a research are very necessary. Glycosylation, it's irreversible post-translational modification. It's important not only for prokaryotic and eukaryotic organisms, but also it's very important for viruses. we can differ two main types of glycosylation, the N-linked and O-linked glycosylation. Here you can see 6 different substances which are actually inhibitors of the N-linked glycosylation, and they affect at 4 different stages of that process. I use them during my laboratory work. Here you can see how many N-linked glycosylation sites are present in the SARS-CoV-2 structural proteins. So the main purpose of my work was to analyze the glycosylation process of structural proteins and its inhibition, but also I wanted to check how the ability of the spike protein to bind to the ACE2 receptor after treatment with glycosylation inhibitors change. So I started my work from the cytotoxicity analysis, and here you can see the results, that at the highest concentration of used inhibitor, the highest cytotoxicity shows monenzin, the lowest miglustat. Rest of the inhibitors shows cytotoxicity between 10 to 30%. After that, I had to create the plasmids thanks to which I would be able to produce those proteins. And I did necessary experiments and I obtained the final constructs, which are pcDNA 3.1 with gene encoding one of the structural protein connected to the Hibit tag. Hibit is a very small tag, which sequence you can see here. And here is agarose gel electrophoresis when I wanted to confirm that I obtained the correct constructs. And in each case, you can see a specific band characteristic for the construct. Of course, I prepared my plasmids for the sequencing to confirm that there's no significant difference which would affect further my further work. So after that, I wanted to check how the tunicamycin affect the production of those proteins. Also, I wanted to check if those proteins actually have the N-glycans. And for this purpose, I did the PNG-ACE assay. And here you can see that minus means not cleaved and plus means cleaved by PNG-ACE samples. And in case of E and M protein, after the PNG-ACE assay, there is no band characteristic for the glycosylated form of this protein. After adding the tunicamycin, the bands are present, but they are definitely weaker in comparison to those one. In case of the N protein, actually there's no difference. After that, I wanted to check how those inhibitors affect the glycosylation in case of the spike protein, the protein which has 22 sites of N-linked glycosylation only in a monomeric form. So here you can see the Western blot analysis of the cell medium. And generally, in case of tunicamycin and NGI1, you can observe a specific shift, which means that bands are lower in comparison to those one, which means that the higher forms of the spike protein, which were there presented, are partially glycosylated. In case of the rest inhibitors, actually there is no significant difference. The same thing I did for the cell lysate. And here also you can observe a shift in case of tunicamycin and NGI1, here and here. And also what's important, here you can observe a non-glycosylated monomeric form of this protein. Additionally, in case of those 3 inhibitors, you can observe a partially glycosylated monomeric form of the spike protein. What's important, those non-glycosylated and partially glycosylated forms were not present in the cell medium. After that, I wanted to check the ability of the spike protein to bind to the ACE2 receptor after treatment with those inhibitors. And here you can see the general rule of this experiment. So there was a SMB domain which was added to the spike protein, and this domain can interact with LGB domain which was attached to the ACE2 receptor. So when those domains can interact with each other and when we add a necessary substrate, we can observe the luminescence signal, which we can easily measure. And here you can see the results. I analyzed the cell medium and cell lysat, and generally you can see that all inhibitors decreased the ability of the spike protein to bind to the ACE2 receptor, and the best results shows tunicamycin and NGI1. So the main conclusions from my project are that the compounds exhibit cytotoxicity in the range of 10 to 30%, of course except the monenzine. The E and M proteins possess nicked glycans, and tunicamycin partially suppresses their formation. Also, the N protein probably lacks N-linked glycosylation sites. The tested compounds inhibited N-linked glycosylation in case of the spike protein, with tunicamycin and NGI1 showing the best efficiency. And also the analyzed inhibitors, by affecting the glycosylation process, decreased the ability of the spike protein to interact with the ACE2 receptor. So here my results may serve as a preliminary data for further necessary research on the structural SARS-CoV-2 proteins. Thank you for your attention. Here are my sources, and I'm ready for your questions.
Thank you, Eva.
Why did you choose such hybrid tag?
Okay, I choose this tag because it allows me to identify the protein not only by the antibodies, but also by the luminescence signal. So there are two ways to identify the protein, thanks it. And also the antibodies are very, very effective.
Okay. So thank you, Eva.
Thank you.
The next presentation will be about optimizing tick-borne encephalitis virus sequencing procedure, a study on Polish Walls reservoir.
Yes.
With Bartosz.
Yes, so my name is Bartosz Mont, and the title of my work is Optimizing Tick-borne encephalitis virus sequencing procedure, a study on Polish Walls reservoirs under the supervision of Dr. Łukasz Rąbalski in laboratory of recombinant vaccines. So the aim of the projects are to determine a prevalence of TBE and phylogenetic diversity from Walls samples, a confirmation that TBEV can be transmitted from pregnant mother to fetus, and updating of PCR's primer spores that are able to perform whole TBEV genome sequencing on Polish virus isolates, and the walls are in Polish Nornice, and this is how they look like, so cute creatures. Tick-borne encephalitis virus belongs to the Flaviviridae family. It is a positive RNA virus, which is important in my work. It is distributed from Central Europe to Far Eastern Asia, and approximately 10,000 to 12,000 worldwide clinical cases of tick-borne encephalitis are reported each year, but about 70% of infections is not reported due to flu-like symptoms. Also, untreated infection of TBEV may lead to meningitis and death, and the death ratio depends on which subtype will focus. So plan of my work was to obtain of primers for PCR and validation with positive controls. Primers I obtained from the publication, then reverse transcription, because this is the RNA virus, entailed amplicon PCR, then primers pool testing, sequencing library preparation and nanopore sequencing, and de novo whole genome assembly and establishing of phylogeny. So nanopore sequencing, this is the platform which was chosen for next generation sequence procedure. The pros it is relatively inexpensive, it has short sequencing time, and it was used in SARS-CoV-2 sequencing in our laboratory, so the laboratory has experience in sequencing viruses using this method. The cons are the limited sequencing accuracy, but for my work it is enough. And this is how this scheme looks like. And there is a membrane in which is nanopore combined with the motor protein. In one side of the membrane is a negative charge, in the second is positive charge, so there is a flow through the nanopore. And when the DNA goes to the motor protein, it is unwounded. One strand goes off and one strand goes into nanopore. And because every base has its own characteristic drop, the program knows which base is going through the pore in that particular moment. So the workflow was the RNA isolation, which I couldn't do because it has to be done in BSL-free laboratory. So I'm starting with RNA, so I have to do reverse transcription to obtain the DNA. After this, I have to perform PCR to amplify my DNA, then quality check as gel electrophoresis, measuring DNA because into sequencing I have to have at least 60 nanograms of the DNA, then library preparation. I obtained from the publication 37 pairs of primers, and the applicants which were obtained after the PCR should overlap whole TBEV genome. So I can say that the TBEV genome were divided into 37 parts. That's the first thing. The second thing that I used two approaches. So first approach was to divide those 37 pairs of primers into two groups. And the second approach was to divide them into 9 groups called pools. That's the second thing. The third thing that this was the tailed amplicon PCR. And it means that the amplicon obtained using the pair 1 of the primers is overlapping part of the amplicon obtained using the primer 2 pair, and so on. So the part of the 2 amplicon is also overlapped by a part of the amplicon number 3. So first results were the initial testing primers, and the validation of method was performed using samples from this military institute. And I did the gel electrophoresis as a quality control, and here are the amplicons. So after this, I performed sequencing, and here are the results. So the most important thing in the screens is the blue area, because the blue area is the coverage. So it means that the higher it is, the better, because it means the more reads from the sequencing were obtained. This is the first criterion. And the second criterion is to have no gaps in this blue area, because it means that the sequence in this gap is not sequenced. Here, for example, it is a decrease, but there is a little bit of blue, so there were some reads. And I can say that in these 2 samples from the Military Institute, I obtained whole genome sequencing, and this is the third sample. It was from the medical university. It should be a positive control, but something went wrong. But they said that, okay, we can move forward. So first results from the Vols samples. Again, gel electrophoresis as the quality control. And here are the results from the sequencing. As you can see, the coverage is low, so there were not too big amount of the reads. And also, there are many gaps. So it was not good sequencing, and I've changed the approach because this is an approach from 2 groups, like dividing the primers into 2 groups. So here it is approach with 9 pools. Again, gel electrophoresis as quality control. Here are the amplicons, and this time sequencing were better. There were bigger amount of the reads, but still in some places, some of amplicons are missing, so I have to deal with them somehow, with these gaps. So this is how I am dealing with it. For example, in this screen, you see that there is a gap where should be amplicon obtained using the 6 pair of the primers. So now what I'm doing is to taking left primer from the pair 5 and pair it with primer right from the pair 6. And also the second pair is left primer from pair 6 with right primer from the pair 7. So I'm trying to obtain the amplicon in this area. The amplicon should be a little bit bigger, about 50%. For now, I did the PCR, but I'm waiting for the results from the sequencing. And here's the discussion conclusions. So, as you could see, the primers used were suitable to the samples from the Military Institute, but not to this from Medical University. And one reason maybe that the viral titer is too low in the evolved samples. That's the first thing. The second thing is that in evolved samples, the viral RNA can be degraded, so the primers cannot attach. And the third possibility is that the regions for design primers poses SMP, so single nucleotide polymorphism that enable off-tate amplicon procedure. So for now, I'm doing further researches and waiting for the results. And during my research, I hope that it will be enough time to design a new set of primers that can be used for whole TBEV genome sequencing in these evolved samples. And that's all from me. Thank you for your attention.
Thank you, Bartosz. Question?
So first, thank you for your presentation, and thanks to your picture, I know that voles are cute creatures, but I'm curious why you choose this animal.
Why? Because of three reasons. First of all, they are the most common rodents in European forests. That's the first thing. The second thing, that they are closely related to human beings than ticks. That's the second thing. And the third thing is that the prevalence of TBE virus is much higher than in ticks. So in ticks, prevalence is about 1%. It means that out of 100 ticks, there is one tick which is infected by TBE virus, and in voles it is about 10 to 20%, so it is much higher and much easier to obtain the TBE virus from the voles.
Okay, thank you.
So I was wondering, maybe I've missed it, but once you succeed with your sequencing, what are you going to do with the data?
A phylogenetic tree, that's the thing. I have to do the phylogenetic tree to see which strains and which variants there is of TBEV, but for now I cannot do it because I'm lack of the whole genome sequencing, and now I'm trying to sequence whole genome from the vole samples.
Okay, thank you, Bartek.
Thank you.
And next presenter will be Kamila Stanek. With her topic is functional and structural analysis of the HCP70 protein, which is common ancestor of the S protein, SSQ1, SSC1.
So, welcome everyone. My name is Kamila Stanek, and the title of my master thesis is Functional and Structural Analysis of the HSP70 protein, the common ancestor of the yeast proteins SSC1 and SSQ1. My supervisors are Professor Rafał Duszkiewicz and Marcin Pitek. So, heat shock protein 70 is a chaperone protein which has many various functions, like protein folding, protein disaggregation, and the regulation of protein activity. But the function I was interested in my project was the biogenesis of iron-sulfur clusters. So what are iron-sulfur clusters? Those are prosthetic groups of many important proteins that are responsible for oxidative phosphorylation, cophotosynthesis, DNA and RNA metabolism, or regulation of gene expression. Alteration in the biogenesis of those clusters can result in many serious diseases. So the HSP70 protein is built of NBD, nucleotide binding domain, and SBD, which is substrate binding domain. And SBD is composed of alpha subdomain and beta subdomain. So in the iron-sulfur cluster biogenesis, the J-domain protein guides the ISU protein, which carries iron-sulfur clusters, to the HSP70. And SBD of HSP70 binds to the LPPVK motif on the ISU protein, and it results in the transfer of this iron-sulfur cluster from the ISU to the acceptor protein. So at the same time, the ATP hydrolyzes to ADP, and later ISU can be released and ADP can be exchanged to ATP by the nucleotide exchange factor. So interesting thing happened in the yeast evolution. The HSP70, the gene encoding HSP70 protein underwent duplication, and those two genes evolve in two different waves. So the result is that now in yeast there are two proteins, SSC1 and SSQ1. So SSC1 protein is kept all the functions typical for HSP70, but SSQ1 got specialized in the iron-sulfur cluster biogenesis. So those two proteins have two different specificities. So to explain why there is a difference in this specificity, the members of our laboratory decided to reconstruct the sequences of the ancestors of those two proteins. And it was my protein of interest in my study. It was NCQ, which is a common ancestor of SSC1 and SSQ1. And on my project, I was working with only with SBD domain of the NCQ. So the objective of my study was to determine the binding mode of the LPPVK peptide from ISU protein, which I mentioned before, to the substrate binding domain of NCQ by the method called X-ray crystallography. So at the beginning, I overproduced the SBD in E. coli. Then I purified it by affinity chromatography. When I had my protein, I had to verify if it interacts with the LPPVK peptide at all. So I performed titration. I titrated fluorescently labeled LPPVK peptide with a SBD domain, and I measured the anisotropy. So the results showed that the peptide interacts with SBD. So I could move on and do the crystallization screening. So I tried to crystallize my protein in few different conditions, in many different conditions actually, in many different buffers. And as a result, I was able to obtain some crystals, and those are microscopic photos of those crystals. Later, I collected those crystals and sent it to the synchrotron in Hamburg, in Germany. And there I collected the diffraction data, and the best resolution I obtained was 2.5 angstroms. So it was good enough to process the data and build a model and determine the binding mode of the peptide. So this is my final result. In one cell unit, I had 2 SBDs. This is the zoom on one SBD. This is the alpha helical subdomain, alpha helical lead, and this is beta subdomain. And here you can see the peptide. On this representation, you can see zoom on beta subdomain with a surface of the protein. And on the last picture, you can see how exactly the peptide binds to the protein. So according to my data, the most probable binding mode of the peptide is in C to N direction with a second proline, which is this one, exactly in the middle of the binding pockets. So what's encouraging is that this result is similar for the results obtained for HSCA protein, which is bacterial HSP70 protein. So that would be all from me, and thank you for your attention.
So you mentioned that SSQ1 has this ability to induce iron-sulfur biogenesis function. Anyway, what about SST1?
Okay, so in fact, both of those proteins are able to take part in the iron-sulfur cluster biogenesis. It's mainly SSQ1 that does it. But SST1 also interacts with LPPVK peptide because I was comparing my results for NCQ protein to the results of the similar tests that were performed but for SST1, and it interacts in the similar way, SST1 like NCQ, so yes.
Thank you, Kamila. And next presenter will be Jan Maciejko with biochemical reconstruction of the cysteine desulfurase ferrodoxin complex participating in the biogenesis of iron-sulfur clusters.
Good afternoon, everyone. My name is Jan Maciejko, and today I would like to talk about biochemical reconstruction of the cysteine desulfurase ferrodoxin complex participating in the biogenesis of iron-sulfur clusters. And iron-sulfur clusters are prosthetic groups of protein which are present in every cell of any living organism, pretty much. They are composed of several iron and sulfur atoms. Most often it is 2, 3, or 4 of each of those atoms. And even though both of those elements are cytotoxic on their own, when combined into a cluster, they obtain completely new properties. And since iron-sulfur clusters are highly evolutionarily conserved, it means that they must perform an important function in the cell. And indeed, they are essential for the functioning and overall survival of the cell because they are involved in a number of intracellular processes such as DNA ribosome assembly, electron transport, or protein catalytic activity. But iron-sulfur clusters do not develop spontaneously. They require an entire cellular assembly machinery. There are a few of such systems, but one of them specifically is responsible for the biogenesis of the majority of iron-sulfur clusters, and it is the ISC system, which is composed of 4 proteins. It is ISCU, which is a scaffold on which it is built, then cysteine desulfurase, ISCS, ferredoxin, FDX, and frataxin. And now, because of the importance of iron-sulfur clusters and severe implications of any dysfunctions in their biogenesis, we want to learn their whole assembly process on a molecular level. Until now, detailed structural analysis was performed only for the cysteine desulfurase ISCU complex. However, recently there have been published results discovering an interaction site, ferredoxin's interaction site on cysteine desulfurase. This indicates that ferredoxin could potentially form stable complexes with cysteine desulfurase, and the goal of my project is to obtain a cysteine desulfurase ferredoxin complex in sufficient amounts for further structural analysis. And in order to do that, I had to overproduce and purify cysteine desulfurase, ISCU, and ferredoxin, then assess whether they are properly folded, and verify whether they form complexes with each other. After a long and labor-intensive process, I managed to obtain highly concentrated purified proteins, and then this allowed me to verify, to analyze their structure with circular dichroism spectrometry. I did it by comparing circular dichroism spectra of my obtained proteins to the spectra of, to known spectra of properly folded ones. And the spectra I obtained exhibited the same shape as the previously reported proteins, spectra of proteins, which confirmed that the whole process of protein purification was successful. Now, the next step was to incubate proteins together to verify their complex forming capabilities. And I did it with the use of size exclusion chromatography. I first incubated cysteine desulfurase and ISCU as a way of method validation because they are known to form complexes with each other. And this is exactly what I observed. Compared to the absorbance peaks of individual proteins, the absorbance peak of a mix of those proteins visibly shifted, indicating that a larger compound, in this case a protein complex, has been formed. But when I incubated together cysteine desulfurase and ferredoxin, the absorbance peak did not really shift, it's only increased. And now, because ferredoxin is fairly small protein, and most probably because of the way it interacts with cysteine desulfurase, the resulting complex doesn't differ much in shape to the original cysteine desulfurase molecule. And that is why I didn't observe any shift on the chromatogram. So to confirm that theory, I denatured fractions eluted during size exclusion chromatography and performed gel electrophoresis on them. And the results showed that ferredoxin was present in the same fractions that the cysteine desulfurase was present in. And all of this led to a conclusion that cysteine desulfurase must form some sort of complex with ferredoxin, which has been pretty much the goal of my project, right? Now, developing a method of obtaining cysteine desulfurase ferredoxin complex means that I can now focus on preparative isolation of that complex, which would further allow me to analyze the structure and specific interactions between those two proteins. Thank you.
Questions?
My question is, have you maybe calculated the molar ratio of this protein in the predominant complex?
Yes.
Because, can I finish?
Yes.
Because your size exclusion chromatography peaks are pretty wide, so it may be that you do not see the complex, but you there is a coelution of these two proteins, and it may not necessarily mean that this is a complex. So maybe you can have another method to confirm that you actually have a complex formation.
Well, I used the excess of substrate ISCU and FDX, which, I mean, it is obviously a good idea to confirm it some other way, but those plots I specifically shortened them so that they don't take this much space. But normally on a plot that has full length of this x-axis, they do not appear all that wide. They are quite narrow. So I think it's just a matter of graphic mistake.
Anyway, on your gel, it seems like there is much more ISCS than FDX.
Yes.
So can you, if you compare the molar ratio, you would see the stoichiometry, or is it that only maybe half or 10% of your ISCS is incomplex, yeah? And you cannot distinguish between ISCS incomplex and ISCS alone.
I mean, that might be the case because, yeah, clearly only a small part of ferrodoxin bound to the ISCS, but still, I mean, it is entirely possible that only like some part of it formed a complex and the rest of ISCS is unbound. But yeah, it would require further analysis to verify that.
Hi, exciting results. I was wondering, have you ever tested if all three proteins can interact?
Yes,
ICS,
yes, indeed, I tested that, but the results were inconclusive because basically the chromatograms looked very very similarly to results of ISCS ISCU complex. So when I performed gel electrophoresis, there appeared both FDX, ISCU, and ISCS those how do you call bands, but I wasn't able to verify whether it is a triple complex or two separate double complexes, because it could be as well ISCS, ISCU, FDX complex and two separate ISCS, ISCU and ISCS, FDX complexes. And I so far didn't verify whether it is like a single triple complex or two separate.
So how will you do it?
I didn't plan it yet. So far I focused on it, but well, there is a way to try to isolate the whole eluted faction and then maybe perform mass spectrometry to verify that. There are several ways we could approach that.
Thank you so much, and now it's time to break, and we come back here for 30 minutes.
Okay, hello everyone. Thank you for being here. So first we have Martina Ivanets. Please, Martina, the stage is yours.
So good afternoon, everyone. My name is Martina Ivanez, and today I would like to present you my master's project called Optimization of Ultrasonically Assisted Proteomic Sample Extraction and Digestion. The experiments were conducted in the mass spectrometry laboratory under the supervision of Dr. Katarzyna Macur and Natalia Musiał. Mass spectrometry is a leading technique in proteomics. It allows us to identify and quantify proteins with a high specificity. Before the MS analysis itself, there are a few sample preparation steps that need to be completed in order to obtain the high-quality MS spectra. The sample preparation steps include the cell lysis and the protein extraction, multiple washing steps to get rid of the detergent and the salt contaminants, and the protein digestion. In my project, I focus on the steps of the protein extraction as well as the protein digestion. Protein extraction is an important step of sample preparation, which is responsible for maximizing protein recovery in yield. To obtain the unbiased coverage of the proteome, this step should be optimized for each sample type. On the other hand, the protein digestion is a crucial but a really lengthy step. The digestion with trypsin can last up to 18 hours, which is an overnight incubation, which makes the whole sample preparation a really time-consuming process. In my research, I tried to optimize the protein extraction to reach the highest number of identified peptides and proteins, as well as to accelerate the protein digestion step. I tried to reach those goals with the use of the ultrasound energy. In my project, I tried to verify two hypotheses. The first one being that the ultrasound energy can lead to more efficient protein release during the extraction, and the second one being that the ultrasound energy can accelerate the enzymatic digestion process from overnight to just few minutes. To investigate the effect of ultrasound energy on different sample types, I used the Rutella ornithinolitica MF1, which is a bacterium that belongs to the Enterobacteriaceae family. It's a gram-negative bacterium. The MF1 strain was first isolated from the gut of the earthworm Dendrobena veneta, and the MF1 genome was just recently first sequenced and analyzed. The other sample that I was working on was the human salivary gland tissue. The tissues were obtained after the parotidectomy procedure at the Medical University of Gdansk. So the whole project was divided into two major sections, the first one being the optimization of the protein extraction, and the other one the optimization of the protein digestion. The optimal conditions were chosen based on the highest number of identified proteins and peptides, as well as the highest protein sequence coverage. So the first step of the optimization of the protein extraction was the selection of the lysis buffers and the sample quantity. Here you can see the graph of the general procedure of the protein extraction. It involved 2 rounds of sonication. You can also see the lysis buffers that were tested. We tested 2 different sample buffers. The first one, the SDS buffer and the RIPA buffer. The SDS buffer was tested in 2 different concentrations, and the RIPA buffer was tested in 2 different modified conditions. So to the first results, here are the results only from the Rutella ornithinolitica MF1. We can see, looking at the Venn diagrams, that the number of identified proteins in comparison, if we compare the SDS and the RIPA buffer, in SDS we can see almost the 13-fold increase in the unique proteins identified. If we investigate further, if we compare the SDS 1% to 3%, we can see the slight increase in the unique identified proteins for the SDS 1%. If you look now at the violin plots, which represent the protein sequence coverage, the wider the violins, the more proteins are on the certain percent coverage level. If we compare the RIPA buffer and the SDS buffer, we can see that the widest violins are at the 0% coverage, which means that the treatment with the LISA buffer did not lead to identification of as many proteins as we have with the SDS. Here is the summary of the number of the proteins and peptides identified. As well, we can see the percent of the miscleavage in every sample. So the best results we obtain after the treatment with the SDS 1% using the 25 microliters sample quantity. So the second step of the optimization of the protein extraction was the adjusting the amplitude of sonication. We tested 4 different amplitudes and compared it to the controlled no sonication. And the last step was adjusting the time of the sonication cycles. So here are the initial results from all of the optimization steps. I just want to highlight that those are the initial results, since we just wanted to see if the ultrasound energy can positively affect the protein extraction. Some of those results were inconclusive, since when we compared the groups, the results there was no such difference. So this whole optimization process needs to be further optimized and investigated. The last part of my project was the optimization of the protein digestion. So I compared two different versions of the digestion, the version 1 being the protein digestion during the sonication for 15 minutes with no further incubation, and the version 2 being the 15 minutes digestion during sonication plus the overnight incubation in 37 degrees. So here are the results for both Rutella and human salivary glands. We can see that the best results we obtained after just 15 minutes of sonication, 15 minutes of digestion with no further incubation, which suggests that not only we managed to accelerate the digestion process, but also we obtained the highest number of identified proteins. So to conclude, the use of the ultrasound energy has the potential to improve the protein extraction in the proteomic workflows, as well as ultrasonically assisted protein digestion successfully accelerates the digestion process from overnight incubation to just 15 minutes and results in higher number of identified proteins and peptides. Thank you. I want to thank you, the whole mass spectrometry lab, for the support during this whole project, and thank you for your attention.
Thank you. Is there any question?
Thanks for a nice talk. I was wondering whether this ultrasound treatment doesn't have any side effects, like miscleavage or something. Have you tested it?
Well, the ultrasound, too harsh conditions can lead, of course, to denaturation and not recovery of the proteins. That's why we tested different times, different cycles. Well, the ultrasound itself is really popular in the proteomic workflows on the different steps, like alkylation, the speeding up the alkylation, and there are also protocols for the digestion. So I don't know what else to say.
Thank you for your nice presentation. I was just wondering, why do you think that the treatment with RIPA buffer leads to higher miscleavages?
So the RIPA buffer in general, the RIPA lysis buffer, is used for the cultured mammalian cells and the soft tissue. It's a mixture of ionic and non-ionic detergents. So my guess would be that the RIPA buffer did not deal well with those sample types that we were using and did not make the cleavage sites for trypsin accessible enough. So that's why the higher miscleavage.
Thank you. Thank you for your nice presentation. Now we have Yulia Karashinska. Yulia, please.
Okay, hello everyone. My name is Julia Karashinska. I perform all research at the Laboratory of Structural Biology, headed by Professor Szymański, and my master thesis is titled Structural Characterization of Protein Interaction Involved in Human Mitochondrial Genome Repair and Maintenance. So each of us has a second set of genes, and the set belongs to mitochondria, the powerhouses of the cell. The smallest chromosome has 45 million base pair, and it happens to be chromosome 21, while mtDNA has just 16,000 base pair and code 13 protein pivotal for energy production, and mtDNA operates in a very challenging environment. It is predicted that mutation rate per base pair is around 10 times greater compared to nuclear mutation rate. So let's now all imagine that one circle represents mtDNA. So it is present obviously in multiple copies, and it might happen that you have a mutation in your mtDNA, but it doesn't have to be present on every single copy. However, the accumulation of mtDNA mutation or damage can lead to the cell dysfunction, and it is connected with the cancer, diabetes, neurodegenerative disease, neuromuscular disorder, and also with aging. So our defense is a robust DNA repair mechanism, and in mitochondria, the primary repair mechanism is the base excision repair. This process can be elucidated in 5 basic steps. steps, starting with the DNA glycosylase, which removes damaged base, leaving a basic site. Then the endonuclease APE1 performs the incision and the DNA backbone. Following that, ExoG removes a basic site, leaving a 2-nucleotide gap, which can be filled by next enzyme, polymerase gamma, and in the final step, ligase-3 seals the nick. According to the latest knowledge, this enzyme cooperate with each other, and they are forming the complex known as mitochondrial repairosome. And what we know about this complex in context of the basic fusion repair. So first of all, key enzymes forming the complex has been identified. The biological importance of this process is clear. We know that MTBR operates through channeling the substrate, and we know that in mitochondrial lysate, ExoG forms complexes with other repair enzymes. What we don't know is the nature of molecular mechanism of complex assembly. The interaction of individual proteins within the repairosome haven't been characterized yet, and also how the regulation of complex assembly looks like. So to address that question, we have to study interaction of proteins within the repairosome. And here, finally, comes my master thesis, which is focused on that part where AP1 and ExoG are cooperating with each other. And the first goal was to isolate the complex of AP1 and ExoG, and the second goal was to study its structure. So the major player in my thesis are AP1 and ExoG. AP1 is a monomer, is present both in mitochondria and nucleus, while ExoG is a dimer. It is present exclusively in mitochondria, and it is a membrane protein. So to achieve the goal, first I perform overproduction of all of the proteins, including truncated mutant and inactive mutant. Then I purify them using metal affinity chromatography, ion exchange chromatography, and size exclusion chromatography. Then we can move on to the first goal, which was fully achieved. So the complex was isolated using ultracentrifugation in glycerol gradient. For this purpose, I prepared 3 different samples containing AP1 alone, ExoG alone, and the complex. And after ultracentrifugation, the sample was fractionated, and each fraction was verified on the SDS page. AP1 was detected with Kumasi blue staining, while ExoG was, since it was labeled ExoG, the fluorescence detection was applied. And this method allows us to separate the complex based on molecular weight of the proteins. That's why we can observe the clear shift in the fractions where APE1 is present comparing to the control. Okay, the second goal was to study the structure. The method which I applied was X-ray crystallography. So first I prepared the complex, then I tested various crystallization conditions, I monitored the crystal growth, and then finally I collect the X-ray data. And then by obtaining a diffraction pattern based on electron density map, I built the atomic model. And here is not to read, just to show you some of the examples of crystals I obtained and data set I collected. And it turned out that these data sets provide us only the structure of ExoG. However, the resolution 1.6 stands for very high resolution, so I continue with this model building. So the experimental data didn't provide us the structure of the complex. That's why I applied different method. So AlphaFold prediction was applied, and by analyzing multiple models and interaction between the proteins and contacts between AP1 and ExoG, I identified some possible amino acids which might be important for this protein-protein interaction. So to sum up, first, the complex was isolated using ultracentrifugation glycerol gradient. Possible amino acids involved in the protein-protein interaction were detected using AlphaFold prediction. And experimental data provide us only the structure of ExoG. So this project will be continued, and what we can do is to isolate more protein complexes within the repairosome and then repairosome itself. Then we should verify AlphaFold prediction experimentally. And I would continue working on obtaining the experimental data to provide the structure. And I would like to mention that I have a great opportunity to present this data on the EMBO conference last week at the poster session. And finally, I would like to thank entire team, my supervisor, Professor Szymański, my lab mentor, Piotrek Purzycki, and you for your attention. Thank you very much.
Thank you, Julia. Is there any question?
why did you choose the method of glycerol ultracentrifugation to purify the complex? Why not some simpler method like size exclusion chromatography?
So that is correct, that size exclusion chromatography is a very common method to isolate protein complexes, and is absolutely correct. However, it cannot be applied to every single complex, and in this case I work with the very dynamic complex, which is part of this reperosome. That's why it just didn't work on the size exclusion chromatography, but I was able to find other methods like this ultracentrifugation to isolate it. So I think that's my answer.
Thank you. Okay, thank you, Ilya.
Thank you.
So next person is Katarzyna Paniak.
Oh, I think you have to switch to my presentation. Okay, good afternoon, everybody. My name is Katarzyna Paniak, and me and Julia, we are a part of a bigger project focusing on the characterization of interaction involving human mitochondrial genome and maintenance. I'm covering a biochemical part. Let's start with the background. So human has two genomes, nuclear one and mitochondrial one. In mitochondria, we have many copies of empty DNA, but they are constantly challenged by reactive oxygen species created during ATP generating processes. Because we have many copies of empty DNA, previously it was thought there's no repair system in mitochondria, yet now we know that one is responsible for the repair of small DNA modifications created by oxidation and base excision repair. So mitochondrial base excision repair is completely independent from the nuclear one, and it's a cascade of enzymes. So here we have the glycosylase that is recognizing the damaged base, is creating a basic site for the AP1, then AP1 is cleaving the backbone, creating a substrate for XOG, and then XOG is creating a bigger gap for polymerase gamma that can actually fill it, and the last step is ligase 3, which is ligating the backbone of DNA. So even though we know the individual effect of each enzyme on the substrate, we don't know how they're cooperating. And it is very important to know how this pathway works, because the accumulation of empty DNA can lead to development of muscular dystrophy, Alport syndrome, Parkinson's, and many more other diseases. So my project is focusing on AP1 and XOG. So my aim of the study was to gain understanding of cooperation between XOG and AP1 during base excision repair. And to examine this, I had to purify the proteins, and later I checked their activity in vitro in biochemical assays with DNA substrate with internal THF, which is like a synthetic analog of a basic site. And that's what is covering my hypothesis. So XOG cooperates with AP1 at a basic site of the damaged DNA. So first, short introduction to purification steps. So all the protocols were established in our laboratory. And for AP1, we perform a metal affinity chromatography, then cation exchange chromatography, and size exclusion chromatography. And this is the result from the final sample in the middle lane. And for XOG, because during metal affinity chromatography we're using urea, we had to introduce the step of refolding, so the activity return. And then later the size exclusion chromatography. And this is my final protein. So now the biggest part of my master's thesis, biochemical assays, we use Phage urea gels in denaturated conditions. So everything you're going to see on a gel, it's actually DNA processed by those enzymes. I did a huge screening of substrate because XOG is a very highly active exonuclease. And finally, we chose a hairpin substrate with internal THF. And here we have the fluorescein. So the signal seen on the gel is going to come from this end. And this is the setup that I also established. So first we did a pre-incubation with AP1, and then later we were introducing ligase or XOG or proteins together. So here we have a 76 nucleotide DNA substrate, and here we have the control with AP1. As you can see, it's cleaving at the THF, so a basic site. And then later I introduced the ligase. So here have the ligase titration, and we have got our AP1 cleavage product and abortive ligation. And what is abortive ligation? Because we still have THF at the substrate, ligase cannot finish its ligation. So it's attaching the 5-adenylate group, which is blocking the DNA repair process. And here is the quantification. As you can see, we have a huge increase abortive ligation and like a minimal proof of complete ligation product. Later, I wanted to check how XOG is working on the AP1 cleavage product. So the setup stayed the same. I just changed the protein for XOG. And here we have the control DNA alone. Here we have the AP1 cleavage product. And here we have the titration of XOG, which is performing very characteristic G-nucleotide cleavage. And this is the most important band for us here, 13 nucleotide, because it cleaved both THF with two other nucleotides. And here we have quantification of that gel. But what will happen if we combine all the three enzymes together? So the setup stayed the same. And as you can see, here is the control of AP1 and ligase. So we can see the abortive ligation product. And then I performed a titration of XOG. And we can see what we saw on the previous gel, the AP1 cleavage product is degraded by XOG, but also abortive ligation product, which is novelty because this function was previously known for aprotaxin and polymerase beta, but this is only present in specific tissues. So here we have the quantification. We can also understand that XOG actually prefers the AP1 cleavage product over abortive ligation, yet it's cleaving both. So to sum up, XOG recognizes AP1 cleavage product and is performing cleavage in such a way that it creates like a perfect substrate for polymerase gamma gap filling activity. It's also preventing accumulation of toxic abortive ligation product. And here we have a scheme when we can see how XOG can recycle the product of abortive ligation back to the correct empty bear pathway. And for the future of the project, I have many ideas, but I think most interesting will be just comparison aprotaxin activity of abortive ligation product with XOG to check which enzyme is primary for this function, and also fully reconstitute the complete bear pathway, so enriched with the polymerase gamma step. Thank you so much, and I would like to thank all the lab members, my supervisors, and for mentoring me and introducing me to structural biology from scratch. Thank you so much.
Thank you. Is there any question?
Yes?
So thank you for the presentation. I have a question because you're talking about the cooperation between the APE1 and XOG, and what's the nature of this cooperation?
Okay, so actually I performed some experiments with the dead mutants of both proteins. So first I introduced the dead mutant of AP1 and XOG wild type, and like I did this experiment also in an inverted way. And I didn't see any stimulation nor inhibition of those proteins on each other, so I think it's strictly substrate product dependent. So I think it relies on the DNA.
Okay, is there any question? Out of curiosity, what was the significance of using THF as a DNA substance in your research?
So it's a stable analog. There's also a natural one that is called DRP, so deoxyribosyl phosphate. I chose this one because of the literature also, and we had checked the activity of XOG that actually can cleave it before in a previous publication done by actually one of my supervisors. So we were based on that, but we also performed some experiments of DRP, but all the setup it got through the whole way of optimization, and it was a pretty long way to get to this point, so we stay with THF.
Thank you, thank you. Thanks. Okay, now we have Eva Izdebska. Eva, stage is yours.
excuse me. Could you please go to the beginning of the presentation because oh, okay, now it's working. Okay. Good afternoon, everyone. My name is Eva Izdebska, and the title of my master's thesis is Generating Mutants of Pseudomonas donguensis strain P482 with inactivated genes related to type 6 secretion system. I worked in the laboratory of plant microbiology under the supervision of Professor Sylvia Jafra. Type 6 secretion system is a molecular mechanism discovered in 2006. It is present in gram-negative bacteria, and it helping them to compete with other organisms. It is built out of 14 proteins that includes Phage, a protein building the spike, HCP1, the one building the main core, and CLP6, the one that takes part in the recycling of the whole mechanism. The base and the tail of this mechanism is similar in build to bacteriophages, and the part anchored in the membrane is similar to other secretion systems. Pseudomonas donguensis is a gram-negative bacteria that is that possesses this secretion system. It is very common, present in soil, water reservoir, and it is a PGPR, a plant growth promoting rhizobacteria, because it can produce hormones, growth stimulants, and also help plants combat their pathogens. P482 is one of the 4 strains of Pseudomonas donguensis, and it has confirmed antibacterial and antifungal properties. The aim of my study is to obtain mutants of P482 with inactivated genes related to this secretion system by interrupting their sequence in the genome by inserting a plasmid inside this sequence. I started my work by isolating genomic DNA from P482 and also selecting a plasmid. At first, I tried to work with PNOC, which is a suicide vector very common in our lab. However, after encountering a few setbacks at the very early stages, I changed my plasmid to be a PRE112. After performing PCR and amplifying the genes of interest from the P482 template, I moved on to enzymatic digestion using enzymes KPN1 and SAC1. I digested both the isolated fragments of the genes of interest and also the plasmid. As you can see, it cuts the plasmid in this area. The ligation in 1:3 ratio led to obtaining a plasmid with an insert, and what is important is that this plasmid carries chloramphenicol resistance. Next, I performed a heat shock transformation into E. coli NM522, and after that I planted the bacteria on selective medium with LB agar and chloramphenicol. In that way, I was able to grow only the bacteria that were able to obtain the plasmid with the resistance for this antibiotic. After that, I isolated the plasmids and performed colon PCR to confirm that everything is all right. I also chose the samples with the highest concentration of this plasmid. And next step was another heat shock transformation, this time to Escherichia coli ST18 lambda peer, because this plasmid has in its genome genes that allow vector transfer that the plasmid PRE112 on its own doesn't have. This strain of bacteria also needs an aminolevulinic acid to grow, so this is another thing I added to the medium for the selection. After that, I isolated the plasmids again, this time from this strain, and performed colony PCR to confirm that I haven't lost the insert at any point. I amplified the fragment between these 2 primers, and as you can see, it is very small. However, if all the steps were performed correctly and the insert was present between these 2 primers, the whole fragment should be around 1,500 base pairs long, as it was confirmed on this agarose gel. The last step was conjugation into Pseudomonas donguensis P482, and this was and confirmation that it was inserted inside the genes of interest. I also performed selection on LB agar with chloramphenicol, but this time without the 5-ALA. In this matter, I was able to isolate only the P482 and get rid of the Escherichia coli. And after that, these mutants were sent to external company for sequencing. After that, I performed comparison between mutants and wild type that includes their growth rate. And this is an example of a growth curve. This one is on LB medium, but the other media were very similar. Results for other media were very similar. As you can see, the wild type, which is this blue one, has a very similar growth rate as the mutants. And as for the shapes and sizes of the colonies, the wild type doesn't differ from any of the mutants. They only differ depending on which medium they were grown on, which is normal. To sum up, I was able to obtain the mutants I was aiming for, and also confirm that they do not differ in any major way when it comes to their growth rate or the shapes and sizes of their colonies or otherwise morphology. I also confirmed that plasmid PRE112 is a better vector than PNOC. And as for the suggested studies for the future, the mutants that I obtained will be able to be used in more antibiosis tests with bacteria and fungi and other common plant pathogens. And maybe they will help to answer the questions like how the lack of function of said genes impacts the functionality of type 6 secretion system, and do the mutants preserve their antibacterial and antifungal properties. These are my sources, and thank you for your attention.
Thank you for your presentation. Is there any question?
So I was wondering, why did you use two different E. coli strains for transformations?
All right. The first transformation was into Escherichia coli and M522 because this type of transformation is, first of all, easier, and also it allowed me to obtain many copies of plasmid in a short period of time. However, this strain doesn't have this whole machinery for vector transfer. That's why I needed another strain, and I chose ST18 Lampapir because it allowed me to further transfer this plasmid into the final bacteria that was Pseudomonas donguensis.
Okay, thank you. Thank you for your nice presentation. Next presenter is Joanna Sasovic.
Can you, oh, maybe, no. Okay. Hello everyone, my name is Joanna Stacevic, and today I'm going to present my master diploma project, during which I was investigating the immunomodulatory effects of venetin-1 in immunosuppressed mice by performing proteomic analysis of spleen tissue. Venetin-1 is a high molecular weight protein sugar fraction isolated from corallomic fluid of Dendrobena veneta F forms. There are already publications about its anti-tumor and antifungal activities, also showing the lack of cytotoxicity against normal vertebrate cells. And most importantly, in case of our project, the recent
in vitro studies on macrophages revealed that Venetin-1 induces the secretion of essential pro-inflammatory cytokines. To further investigate the immunostimulatory properties of Venetin-1, we also use cyclophosphamide to induce the immunosuppression in tested mice. CP is a very common chemotherapy agent and immunosuppressant used since 1959. It needs to be converted into active metabolites, which takes place mainly in the liver. The most crucial metabolite is phosphoramide mustards that forms cross-linkages in DNA via alkylation, consequently leading to cell apoptosis. The most vulnerable to its action are rapidly proliferating cells because their repair mechanisms are less efficient. Another important metabolite, acrolein, is responsible for multiple toxicities because of the production of reactive oxygen species. Now let's move to the methods. There were 4 groups of healthy mice with 3 individuals in each. The first group was a control one, only treated with saline. The second group was a CP control group. Initially, mice were treated with CP to induce immunosuppression and ended receiving saline. The groups 3 and 4 also were initially treated with CP, and after that, mice were administered with Venetin-1 at 2 different concentrations. Now what happened next? Mice were euthanized in order to isolate the organs, including spleen. Then spleen was homogenized and cell lysis was performed. The next steps were protein digestion using fast procedure and peptide purification on C18 solid phase. And finally, we performed mass spectrometry, spectrum registration, and bioinformatic analysis. The statistical analysis showed that treatment with CP induced upregulation of 22 proteins compared to control group, whereas after giving the lower dose of Venetin to immunosuppressed mice, there was a lack of any upregulation. On the other hand, the higher dose of Venetin resulted in upregulation of even larger amount of proteins, especially compared to the CP group. During the functional analysis, I was checking in which signaling pathways these upregulated proteins may assist. In CP group, there was only one upregulated pathway identified, called African trypanosomiasis. According to the literature, this pathway leads to immunosuppression. In case of higher dose of Venetin group, we observed the upregulation of over 20 pathways, some of which are presented in the table. And as you might notice, many of these pathways are linked to immune and proliferative processes. And finally, I also looked for the information about some of the proteins in the different publications. I focused on the proteins which were upregulated only in Venetin-treated groups, and also that had high fold change. As presented in the table, many of these proteins were earlier detected in immune response and inflammation. To summarize, the very first in vivo Venetin study showed its immunostimulatory effects by inducing the upregulation of proteins involved in immune response activation. This finding confirms its potential application in non-specific immunotherapy, and also it makes it potential ingredient in medical preparations that are meant to treat different immunocompromised conditions. Thank you for your attention.
Thank you for your presentation. Is there any question?
I was wondering, maybe you could explain how Vanadin-1 is prepared?
Of course. The protocol of preparation was designed by scientists from Maria Skłodowska-Curie University in Lublin. After collection collomic fluid from Dendrobena veneta by electric shock method, it was centrifuged to get rid of the cells. Then it was sterilized, and the most important part is heating at 70 degrees for 10 minutes. After that, fraction is dialyzed in water using filter with that cuts off compounds larger than 14 kilodaltons, and this high molecular weight fraction is Vanadin-1.
Thank you for your presentation. Also, I have one question. Oh, sure.
You talked about the upregulation of proteins. What about the downregulation?
Yes, of course. In my paperwork, I will talk about also downregulated proteins. These proteins take part in such pathways as neurodegenerative diseases and metabolic pathways. So in case of my project, it had more sense to focus on upregulated proteins during this presentation, but of course I will analyze downregulated too.
Thank you. Thank you for your presentation. So next we have Weronika Siczebek-Remontowska.
Hello everyone. I'm Weronika Siczebek-Remontowska, and today I'm going to present my master's project titled Evaluation of the Influence of Vanadin-1 Nanoparticle on Candida auris cells, conducted in the mass spectrometry laboratory. Vanadin-1 is isolated from the collomic fluid of Dendrobena veneta earthworms, and these annelids are easy to obtain, and they don't require sophisticated cultivation. And because they live in the soil, they have developed various defense mechanisms against microbes present in the soil, such as bioactive substances, including enzymes in their collomic fluid. And additionally, they have been used in the Far Eastern medicine for many years to treat a variety of conditions, ranging from infectious diseases to cancers for many years. Vanadin-1 is a protein carbohydrate fraction isolated by Professor Marta Fiołka from Maria Curie Skłodowska University in Lublin. It is comprised, among others, of lyseinins, which are hemolytic and cytotoxic toxins that can form pores in sphingomyelin-containing membranes. It also has lysozyme-like activity, which means it can hydrolyze peptidoglycan in bacterial cell wall. It also presents antifungal activity. It targets Candida albicans cell wall, leading to apoptosis of disease, but it's not toxic towards fibroblasts. Candida auris, identified in 2009, it's a growing cause of nosocomial infections, especially during COVID-19 pandemic. And it attacks mostly immunocompromised patients with quite high mortality rates. And the vast majority of the isolates are multi-drug resistant, with some isolates being resistant to all classes of known antifungals. And since Vanadin-1 has been proven effective towards Candida albicans, my hypothesis is that it has a similar impact on Candida auris cells. And the goal of my project was to treat Candida auris with venetin-1 nanoparticle, evaluate its influence on disease, while further characterizing the venetin-1 nanoparticle. I want to start with the characterization of venetin-1. And since I mentioned that it has lysozyme-like activity and the presence of lysozyme was previously suggested via Western blotting, I wanted to isolate the lysozyme from the raw colomic fluid and venetin-1. And to do this, the lysozyme-specific antibodies were embedded onto cephalose beads, then incubated with raw colomic fluid, venetin-1 or pure lysozyme, then eluted, digested, and analyzed on a mass spectrometer. And the analysis showed that the lysozyme is present in both raw colomic fluid and venetin-1, alongside lysozyme-related protein 2 and other proteins such as tubulins, histones, and actins, but only as an example, fragment of tubulin alpha chain is present here. To find out if lysozyme was present in previously analyzed samples of raw colomic fluid and venetin-1, the retrospective analysis was carried out, and the Orbitrap mass spectrometry data from raw colomic fluid and venetin-1 previously separated via gel electrophoresis was compared with Annelida protein database expanded with human and chicken egg white lysozyme sequences. And the results show that in both DVR and venetin-1, lysozyme was present, but in the case of venetin-1, the lysozyme was present in only one of the separated fractions. Retrospective analysis was also conducted for the venetin-1 separated on two-dimensional gel electrophoresis. The specific bands were cut from the gel, then digested and analyzed on the mass spectrometer. And the bands that are marked with green show where lysozyme was detected. The bands marked in purple mean there was no lysozyme detection. However, in all of the bands analyzed, the other proteins, such as lysandines and lysandine-related proteins, were still present. And since lysozyme is always present alongside lysandine, we might kind of assume that they can form complexes. And here you can see proposed lysozyme-lysandine dimer and trimer structures proposed by Professor Artur Giełdoń from Laboratory of Simulation of Polymers. And the venetin-1 samples are currently being analyzed in the Solaris National Synchrotron Radiation Center in Kraków. And the results from that analysis will be compared with the proposed models to improve the structure of the complexes. And finally, I want to talk about the influence of venetin-1 on Candida auris cells. And in order to do that, I conducted a 48-hour growth curve monitoring of Candida auris strain CDC B11903 in YPD, treated with 3 different concentrations of venetin-1 in YPD medium. And as you can see, compared to the untreated control, venetin-1 does not really affect the growth of Candida auris in tested conditions. The cells were also observed under the microscope, and compared to the untreated control, cells tested with both 50 and 100 micrograms per milliliter of venetin-1, we cannot really observe any changes in these conditions. In comparison, cells treated with a known antifungal clotrimazole are clumped together and their number is clearly decreased. And here are the concluding remarks of my project. Lysozyme was successfully isolated from raw colomic fluid and venetin-1 via immunoprecipitation. Expanded Annelida database search confirmed the presence of lysozyme, lysandine and lysandine-related proteins in raw colomic fluid and venetin-1. Tubulins, actins, and histones detected alongside those proteins might play a role in stabilizing venetin-1 structure. And preliminary data suggest that lysozyme might form complexes with lysandine proteins. And finally, Venetin 1 does not appear to be effective against Candida auris in tested conditions. I would like to thank all of the following people for their immense help, and thank you for listening to my presentation.
Thank you. Thank you for your presentation. Questions?
Thanks for the talk. I was wondering, since this Venetin 1 is isolated from animals, is it somehow standardized? Have you maybe analyzed several samples or different types of isolations and stuff?
Actually, so far, there is no standardization apart from the fact that the earthworms are grown in the same conditions, and the fluid and the Venetin 1 itself is isolated in the same conditions. But after each isolation, each batch is analyzed via Fourier transform infrared spectroscopy to make sure that the batches of Venetin 1 are similar in the chemical structure. So this is the type of standardization that they do now. Maybe in the future, it will be like a whole protocol of standardization, but not for now.
Thank you. Thank you for your presentation. Next person, we have Kamil Kava. Kamil.
So good afternoon, everybody. I'm here to present my master thesis project, which title says In Silico Selection and In Vitro Verification of the Activity of Novel Tragitopes. And what are those? Those are polypeptides with telerogenic activity. My master thesis supervisor is Professor Tomasz Grabowski, and he is from the pharmaceutical faculty at GUMET, and my additional supervisor is Dr. Jagoda Mierzejewska, and he works at Hirschfeld Institute at Wrocław. And here I wanted to introduce you on why we have picked up on such a project, and it was to aim at the immunomodulation in our organism. All these problems that you can see here on the slide are problems related to either to autoimmunity or immunogenicity, and we wanted to help fixing those problems with immunomodulation. And we decided to go with tragitopes, and the name tragitopes stands for T regulatory cells epitopes. And those are peptides that exhibit very strong ability to bind to MHC class 2, and some of the papers also suggest binding to MHC class 1, but they are being presented on dendritic cells to FOXP3 positive T regulatory cells. For now, I will call them Tregs for short. And when the Tregs are activated by tragitopes, they secrete immunosuppressive cytokines such as interleukin 10 or TGF-beta. And thus, it induces the immunological tolerance. And hence, this is a very rarely touched topic. We wanted to discover new peptides with tragitope activity. And this is how we have done it. For starters, with my supervisor, we have selected 3 candidates for tragitopes. And the name of those stands for 3 first amino acid of their sequence. And those are MCD, LLG, and ILF. We have also selected 2 additional peptides that were the already established tragitopes in literature, and they served us as a positive control. And those are EEQ and FSG. Then I have synthesized them via solid phase synthesis. I have identified them via mass spectrometry. I have purified them, and then I have checked their stability in cell culture conditions that were further used in vitro assays. And this is the basic scheme of how we have conduct our bioassays. And firstly, we have started with setting up the culture of bone marrow cells, the murine ones, and in presence with 2 cytokines that were meant to induce the dendritic cell differentiation. We then have transferred them onto 6-well plates. Then we have introduced our tragitope samples. Then we have analyzed the phenotype of the cells by flow cytometry, and at the same time we started a second co-culture of BMDCs with spleen cells, and to be able to then identify the cytokine secretion and the Tregs population. here is how the flow cytometry analysis was designed, and this particular one was to find the expression of MHC class 1 and 2, and to find the expression of additional costimulatory molecules. This is the very similar scheme, but this time we wanted to find the Treg population. And now that you have a pretty clear vision of what I have done, I wanted to present you the results. And we have started with setting the MTT test to find the cytotoxicity of our peptides. And we did this to establish the further concentration for further studies. And we also decided with which cells we want to go with. And we decided to go with BMDCs, so bone marrow dendritic cells. And here are the first results from flow cytometry. And thankfully, we didn't observe any unexpected decrease in cell viability, which was good. And then we observed actually that various different concentration of all trigotopes have increased the population in dendritic cells, which was our slightly first positive finding. And then we analyzed the phenotype of the cells. Firstly, the MHC class 2, and hence the characteristics of trigotope. We hope that the MHC class 2 expression will be increased. And it was by two peptides, which was FSG peptide, which was our control, and the second one, our research one, the ILF peptide. Then we have also checked the expression of CD86 due to the fact that CD86 with MHC class 2 play the most significant role when it comes to antigen presentation for Tregs. And we could also observe that 3 of the peptides were able to increase the expression levels, and these peptides were FSG, MCD, and LLG. And now that we have established phenotype, we wanted to check the pro-inflammatory cytokine, which was interferon gamma, and the most crucial cytokine for Tregs induction and in general induction of immunotolerance, the IL-10. We have started the checking, and we have observed that the cells treated with ILF peptide showed high increase of interleukin-10, which was very positive for this peptide. And also a couple of other concentration of peptides showed the increased levels, but they were not statistically significant. And on the other hand, interferon gamma was also increased in a couple of cases, but still not in all of the cases where the differences were statistically relevant. And then we could, when we had the established concentration of cytokines, we were able to compare the two of them, the interleukin-10 and interferon gamma, and we were able then to tell by the balance whether the cell response shifts more towards the pro-inflammatory or pro-suppressive activity. And we have almost all of the peptides, instead of the LLG peptide, has been shown to slightly increase the pro-suppressive activity, although these differences were not statistically relevant. And the Treg population was also visibly increased, but again, the statistics says differently. And now that we have seen our results, we can jump to conclusions, and that we were able to select our most promising candidate, that was ILF. And it is due to the fact that it was able to increase the dendritic cell population. It was induced the expression of both MHC class 1 and class 2 on dendritic cells. Then it also highly increased the IL-10 cytokine secretion. And then, although the interferon gamma was also secreted, the balance between the two of them showed the suppressive activity of the trigotopes. And in general, the ILF tends to follow the same trend as the peptides from positive control. And in general, it contains the characteristics of the in theory, they work the same. And the other peptide was the MCD peptide. And although it had a little bit lower impact on cells, it still showed to also follow the very similar trend to ILF and the trigotopes. And thank you very much for your attention.
Thank you. Is there any question?
Thank you. So I have two questions, actually. So the first one would be, what is so specific about those peptides then that they can be considered with this activity? Because obviously in your setup you use splenocytes, so there is no Treg enrichment in this. So why we're expecting this sort of results?
Direct induction?
Yeah, well, when you're running your ELISpot, I believe, then you have like all the culture of splenocytes.
Yeah.
So they're not pre-selected. So that's how we're expecting that they will only be presented to the T-Rex. Or whether we sort of expecting expansion, just the T-Rex. That's one thing. And the other question that I had, yeah, in your calculations of the type 1 response versus Treg response bias, you can see that it kind of is counterintuitive with the dose dependence. So it's like lower dose gives higher, I think.
Yes, that's both very good questions.
Thank you.
And maybe I will start with the second one. And we were wondering why the Treg expansion was so low, and it was contradictory to what we expected. And we had the hypothesis that it is due to the fact that we treated the cells for 5 days. And we want to now to set the timing and to check at the various different days of how then the T cell population comes up, because we expect that we had the secretion of interleukin 10, and probably the Tregs also were increased, but then because of the 5 days, the Tregs were exhausted and the population died. And yes, that's what we suspect, but we have to also check it. And no, we just expect that the EQ at the high concentration, for example, should be the biggest one, because it was shown in the literature. But we expect that the exhaustion was the problem here. And to the first...
What's so specific about them that they target the Tregs mainly?
I'm not pretty sure, but based on the history, in general, they were discovered because the scientists were wondering why therapeutic monoclonal antibodies were pretty tolerable in our organisms. And they checked the Fc region of the antibody, and they have found these peptides there.
Okay, and so they're from Fc parts?
Yes, not only. They're not specified to antibodies, but the first one was discovered in the Fc region, yes. But for example, the FSG peptide, which was also the established trigatopes, I don't remember which virus, but it was from the viral protein.
Okay, but do they have like any in complex with MHC at the class 1? With complex, when they complex with the class 1 or class 2 molecules, yeah, do they have a structure that would indicate different affinity with the TCR?
There's very few papers on this topic. When I picked up this, it was about 30 papers.
Right.
So we don't know yet.
Thank you.
Thank you very much.
Thank you. Thank you for your presentation.
Thank you.
Next, we have Marta Sobolewska. Marta, the stage is yours.
So, good afternoon. My name is Marta Sobolewska. I'm pleased to present you the findings of my master thesis about analysis of dikaiosin P2D1 production by Dikia Dadanti. So, the primary subject of my research is Tylosin, a nanomolecular structure, and this term derived from the fusion of two words, first phage, tail, and the bacteriosin. Tail because of the structural similarity to the bacteriophage tail. They are also encoded in bacteria genomes and released via bacterial lysis. And the second part, bacteriosin, the molecules released by bacteria to compete with others by inhibiting their growth or leading to their death. So, Dikia dadanti is a pectinolytic bacterium which is known from causing a disease of plants, for example, softroot or bacteria blackleg. And recently, it has been discovered that this bacteria also produce tylosin called dikaiosin P2D1. And these tylosins are able to kill few strains of bacteria, which are also plant pathogens. And due to that, this dikaiosin in the future may be a potential plant protection agent. So we already know that these bacteria produce dikaiosins, but we don't know when and how many. And to answer this question, I performed few tests.
regarding counting the number of active tylosin, measuring the gene expression, and titer of viable bacterial cells which producing this tylosin. And I also tested several other compounds if they are able to increase the level of production of those tylosins. So to reach my goals, I used few methods. First of them were tylosin production by adding the inducer into bacteria culture. And after purification, I conduct semi-quantitative spot test in which are determining the highest tylosin dilution which causing bacterial death. And what is important, this tylosin activity correlate with the number of active tylosin particles. So it was somehow counting the number of tylosin in my probes. And I also measured the gene expression by real-time qPCR from designing primers to data analysis. So what is important, my primary inducer in the study were mitomycin C, which is known for activation of SOS response in bacteria. So firstly, I investigated if the concentration of inducer have impact on the production of tylosin, and the answer is yes. The highest production of tylosin were observed in range of concentration mitomycin C between 0.5 to 1.5 microgram per milliliter. And on the right side, you can see how this production of tylosins change over the time. And here is the point where I added mitomycin C in this bacteria culture, and the highest production of the tylosin is observed 6 hours after adding the inducer, and it remains relatively stable. So then to conduct real-time PCR, I designed 8 pairs of primers. 5 of them were complementary to the sequence of genes encoding tylosin-related proteins, and 3 of them were complementary to reference genes. And here are the results of my expression analysis. And you can see that I checked expression of 3 genes, fiber, tube, and sheath. And we can see on the control that this gene expression is relatively stable and do not change over the time, in contrast to the probes which were induced by mitomycin C, and the expression increased after 1 hour, 2 hours, and 4 hours, and the highest expression were observed 2 hours after addition of the inducer. And I also checked if another compounds can increase this level, and the antibiotics like norfloxacin and ciprofloxacin, yes, and also hydrogen peroxide increase this level. But ampicillin and chloramphenicol decrease the level to compare to the control, so the probe without inducer. And here the summary of the induction of tylosin production in the Dickia dadanti cell. Green bars indicate the gene expression, so the highest occurs 2 hours after the addition of the inducer. This line indicates the changing in activity of the tylosin, and the peak is observed 6 hours after addition the inducer into the bacteria culture, which correlates with bacterial death, because this indicates the deaths of Dickia dadanti which produce these tylosins. So to conclude, I established that these 4 substances increase production of tylosin by Dickia dadanti. The highest expression of genes encoding structural elements of tylosin is observed 2 hours after adding the inducer, and also the peak of the tylosin production is observed 6 hours following mitomycin C treatment. And I want to thank my lab team for all the support and their knowledge.
And also thank you for listening to my presentation. Thank you.
Thank you. Thank you for the presentation. Is there any question? Okay, thank you. Thank you. Next person is Zofia Rankowska.
Hello, my name is Zofia Rankowska, and I would like to introduce you to my master project titled The Role of FGFR, ETV4, and Hippo Pathway in Resistance to Therapies of Triple-Positive Breast Cancer. I conducted my research in the Laboratory of Molecular Enzymology and Oncology. Breast cancer is the most common cancer among women worldwide, and it is divided into 4 main subtypes based on expression of estrogen receptor, progesterone receptor, and AGR2. And those subtypes are luminal A, luminal B, AGR2 enriched, and triple negative. In my project, I focused on triple positive breast cancer, which is characterized by high levels of all 3 receptors. And patients with triple positive breast cancer receive worse prognosis than those who are HER2 negative, and they also very quickly develop resistance to drugs. Currently, they are treated with chemotherapy that is combined with anti-AGR2 drugs and then followed by anti-ER therapy. So, tumor microenvironment plays a major role in cancer development. Cancer-associated fibroblasts can activate fibroblast growth factor receptors on cancer cells, thus activating several pathways that lead to cell proliferation, survival, and drug resistance. It's been previously shown by our team that FGFR activity can protect breast cancer cells from anti-ER therapies such as tamoxifen. A preliminary study conducted by our team showed that FGFR signaling increases mRNA level of ETV4 transcription factor in cells that were both treated with tamoxifen and lapatinib and not treated with them. Tamoxifen is an anti-ER drug and lapatinib is an anti-HER2 drug. There is some data connecting ETV4 with Hippo pathway. So Hippo pathway is responsible for cell proliferation and organ size control. And when this pathway is activated, it leads to phosphorylation of downstream transcription co-activators YAP and TAS, which then leads to their degradation and cytoplasmic retention. But when this pathway is not active, YAP and TAS can enter the nucleus where they bind to transcription factors such as TEDs, thus leading to pro-proliferative gene it's been shown that when the Hippo pathway is dysregulated, it can be involved in breast cancer progression, invasion, and drug resistance. And there is also some studies available that indicate that ETV4 activity is regulated by FGFRs and also that ETV4 potentiates YAP retention in nucleus. So taking all of that into consideration, the aim of my project was to verify FGFR involvement in triple-positive breast cancer resistant to therapies and the role of ETV4 and Hippo pathway in FGFR-dependent resistance of triple-positive breast cancer to anti-ER therapies. My hypothesis was that FGFR-dependent induction of ETV4 and Hippo pathway is involved in development of triple-positive breast cancer resistance to anti-cancer therapies. In my project, I worked with a triple-positive breast cancer cell line, BT474, and first I wanted to investigate the influence of FGFR activity on levels and activation of Hippo pathway proteins and on level of ETV4. First thing I observed was an increase in YAP phosphorylation on serine 127, which is responsible for cytoplasmic retention of this protein. And this happened despite of a decrease in upstream LATS phosphorylation, which might imply a Hippo-independent
phosphorylation of this protein. I also observed an increase in ETV4 level, which is consistent with the preliminary mRNA data. I also observed an increase of ETV4 and YAP levels in cytosol after FGF2 treatment, which again is consistent with the phosphorylation from previous experiments, but is contrary to our hypothesis. As I mentioned before, it's been shown that FGFR activity can cause resistance to tamoxifen. I wanted to see if inhibiting YAP-TET would abolish this resistance. So I used two inhibitors. The first one was IK930, which did not abolish this resistance, but a YAP inhibitor, verteporfin, abolished this resistance, which might imply that using YAP inhibitors in the future with anti-ER therapies might improve treatment. And the difference between those two inhibitors, the action of these two inhibitors, can be explained by the fact that YAP not only binds to TETs, but also to other transcription factors. In the second part of my project, I wanted to investigate the role of YAP, TAS, and ETV4 in double resistance to anti-ER and anti-HER2 drugs, tamoxifen and lapatinib. Cell line resistant to both of these drugs was derived in our team, and I did a MTT2D test with different concentrations of both of these drugs, which showed a higher cell viability of the double resistant cell line in all of those concentrations. I also confirmed this with a 3D growth assay in Matrigel, where again the double resistant cell line showed higher proliferation in both drugs. And then using Western blot, I investigated change of YAP, TAS, ETV4, and receptor levels between those two cell lines. And what I observed first was an increase in HER2 and ER level with a decrease in PR level, which is common in cell lines resistant to anti-ER and anti-HER2 drugs. But what is more important, I observed an increase in FGFR4 level with an increase in YAP and decrease in TAS level, which could be the reason for this double resistance, but this needs to be investigated further in the future. So summing up, my conclusions were firstly that FGFR signaling increases mRNA and protein level of ETV4, also that FGF2 phosphorylates YAP in a hypo-independent manner. The next conclusion was that YAP inhibition abolishes FGF2-dependent resistance to tamoxifen, which points to a potential application of YAP inhibitors in combination with anti-ER therapy in the future. And my last conclusion was that the mechanism of VT474 resistance to both lapatinib and tamoxifen is likely based on increased FGFR4 level and inverted YAP-TAS ratio. And thank you very much for your attention.
Thank you. Thank you. Is there any question?
I was wondering what experiment did you use to receive this mRNA data, which you showed at the beginning?
Yes, so it was here. We used the BCP360. It's a panel of around 700 genes that are connected to important signaling pathways in breast cancer, and it helps. It gives us information of the response to different treatments. So in our case, FGF2, tamoxifen, and lapatinib.
Thank you. Is there any other question? Okay, thank you. So we have next attendance, which is online, Alexandra Shtur. Okay, she will join us in a moment.
02 phage has genes encoding encoding virulence-related proteins like protein E and virulence factor SRE, which can cause increasing virulence of the bacterial host when it is integrated to bacterial genome. The aim of the project is characterization of bacteriophages infecting clinical Clostridioides difficile strains obtained from hospitals in the Silesian Voivodeship. So we wanted to check the presence of bacteriophage in bacteriophage CDKH02 in those samples. The materials I used are a collection of 42 clinical strains of Clostridioides difficile from Silesian Voivodeship and PCR primers. First PCR primers was specific to Holline gene fragment of Myoviridae and Syphoviridae families. Then PCR primers specific to TPE gene fragment, which is housekeeping gene in Clostridioides difficile, and three pair of primers specific to unique sequence of the CDKH02 phage. Firstly, we identify prophages in bacterial genome. Then we do phage induction, and in the end, search in this bacteriophage collection to detect the CDKH02 prophage. We culture bacteria in anaerobic conditions in anaerobic chamber. Then we prepare the phage induction by adding mitomycin C to the bacterial suspension. And then we detect of prophages in bacterial DNA. After that, we needed to digest this bacterial DNA and also disintegrate the capsids of adult form of bacteriophages by heat. After that, we obtained the suspension of phage DNA and analyze this. Now we can go through the results. So firstly, the part of analyzing the bacterial genome. So it is before DNase 1 digestion. Here we have signals from a TPI gene, which means which confirm a good condition of bacterial DNA. And here we have some signals from Syphoviridae and Myoviridae phages. So we could assume that in those strains we have some prophages from those families. Here we have basically the same series of reactions, but those experiments were done after bacterial DNA digestion by DNase 1 and capsid disintegration. So here material is phage DNA. Yeah, and that's why we have no any signals from TPE. Here we have some signals from Myoviridae bacteriophages family. So we can confirm that we have some functional bacteriophages from this family. And we can confirm it because a genome of those bacteriophages was protected from DNase 1 by those capsids. Here we have results of PCRs with sequence with specific sequence of CDKH02 phage. Here we have strains which have only one specific sequence, so we can't confirm the phage CDKH02 presence. But in strains 3 and 4, we have all three signals for all three specific regions. And in those strains, we can confirm that those strains has some functional prophages in their genome. Here we have the table with all the results. In orange, I tagged the CDKH02 phage presence. And here in ribotype 955, we have additional confirmation of phage presence by sequencing. Yes, as we can see, the majority of hypervirulent strains has CDKH02 phage in it, in their genome. And as I said, the tests confirm the presence of the phage in hypervirulent strains of bacteria. So I can confirm that phage CDKH02 spreads very well also in Silesian voivodeship. Also, previous research proves that the bacteriophages can increase virulence of the bacterial host, but exact mechanism of this process is unknown and require further research. And I want to mention that my research is part of bigger project under the title "The Role of Prophages in the Virulence of Clinical Strains of Human Pathogen Clostridioides difficile in vivo and in vitro and in vivo." Yeah, this is all for me. Thank you very much for your attention.
Thank you. Is there any question? Okay, I have one question. I have to go upstairs. My question, okay, I'm going to sit here. What does she think about the development possibilities of this project in general? But I'm going to ask this question. Should I go there?
I asked her on the messenger.
Okay.
We have one-way connection.
Okay, so okay. So knowledge obtained in this research can be useful to clinicians and epidemiologists due to the fact we're dealing with the higher virulence strains of Clostridioides difficile. For example, yeah. For example, we can implement some surveillance on infections episodes of Clostridioides difficile. And in the future, we probably can also take advantage of the fact that the phage CDKH02 infects hypervirulent Clostridioides difficile strains, and we use it in phage therapy. Of course, it is important to prepare research to ensure that the phage does not threat the patients by increasing the virulence of the bacteria. So the phage should be modified by removing the virulence-related genes and lysogenic genes. So as a result, we could obtain the lytic phage, which can kill hypervirulent Clostridioides difficile bacteria, which could be a perfect model for phage therapy.
Thank you, and thank you for your attendance. We have 30 minutes breaks, and after that we have the award ceremony. Thank you.
Halo, halo. Działa, dobra. Dobra, no, można trzymać, po prostu podawać tam jak będzie.
Dobra, tak, myślę, że będzie łatwiej. Szanowni Państwo, myślę, że tą część możemy już po polsku, będzie łatwiej, zgrabniej i może mniejszy stres, żeby nie przekręcić jakichś nazwisk. Po pierwsze, ja wszystkim Państwu bym chciała bardzo serdecznie pogratulować. Pogratulować Państwu bardzo dobrych prezentacji. Ja nie siedziałam tu na sali, ale słuchałam online, bo tak mi było łatwiej, bo niestety praca papierkowa w tym roku mnie całkowicie wyłączyła, ale zaglądałam tutaj na salę i widziałam część z Państwa prezentacji, które były naprawdę bardzo dobre. Dziękuję za dyskusję, dziękuję za udział pracowników, którzy zadawali pytania, no ale przede wszystkim Wam Państwu gratuluję, bo to były naprawdę 2 dni fajnego wydarzenia i pokazanie wszystkim, jak dobre są nasze prace magisterskie. To znaczy, jak Państwo są dobrzy, ci, którzy kończą. W tym roku udało nam się z władzami dziekańskimi zrekrutować kolejne firmy, które ufundowały nagrodę. Bardzo się z tego cieszymy, ponieważ nagrody są zacne. Muszę przyznać, że firmy chętnie nas wspierają i fundują nagrody dla Państwa. Myślę, że widać, że są atrakcyjne, skoro tylu z Państwa się na te nagrody zgłosiło. Dobrze, to żeby już nie trzymać Państwa w takim oczekiwaniu i niepewności, to mamy kilka... Tak.
Chciałem powiedzieć, dlaczego firmy się zgłaszają. Dlatego, że Wy jesteście po prostu dobrzy. Jakbyście nie byli dobrzy, to firmy nie byłyby tym zainteresowane.
Więc tym bardziej bardzo się cieszymy. Więc słuchajcie, ja zacznę od naszej nagrody imienia pani profesor Podhajskiej, dlatego że to jest jakby nasza nagroda i tutaj pracownicy na tą nagrodę, na Państwa oddawali swoje głosy, więc w tym roku nagrodę dziekana otrzymał pan Marcin Banacki. Bardzo serdecznie gratulujemy. Proszę się zgłosić później do biura dziekana, tam wszystkie formalności, to jest na razie dyplom, ale formalności będą tak. Dobrze, to już pierwsza nagroda za nami. Teraz mam tutaj na liście nagrodę, którą ufundowała firma Kawaska i ta nagroda w postaci laptopa powędruje do pani Julii Karasińskiej. Serdeczne gratulacje. Serdeczne gratulacje od nas. Dziękuję bardzo. Firma taka bombonierka kocięcia. Tak, do zdjęcia koniecznie. Tu zapraszamy pana prezesa. Super. No i szanowni Państwo, 2 następne nagrody ufundowane przez firmy farmaceutyczne. I teraz jest nam niezmiernie miło, że w tym roku udało nam się przekonać firmę Adamet do ufundowania dla Państwa płatnego stażu. Trochę byliśmy pełni niepokoju, ponieważ no, staż jest w Warszawie, w związku z tym nie wiedzieliśmy, czy się Państwo na ten staż zapiszą i trochę ja tak z duszą na ramieniu najpierw przez parę miesięcy rozmawiałam z firmą, przekonywałam ich, a później tak naprawdę rozmawialiśmy w naszym gronie, co będzie, jak się nikt nie zgłosi. I tu bym wtedy swoim nazwiskiem gdzieś tam musiała się ratować. Natomiast udało nam się i dziękuję Wam za to, że się tam zgłosiliście, a laureatem nagrody jest pani Weronika Kłopotek-Główczewska. Pani Weroniko. serdecznie gratulujemy. I taki piękny właśnie czek. No właśnie, czek jest zacny, też właśnie strasznie nam się podoba.
Dziękuję później.
I ostatnia nagroda, co prawda nie ma czeku, ale jest inna niespodzianka w przypadku firmy Polpharma. Dzisiaj dostaliśmy informację, że firma Polpharma zdecydowała się podnieść wysokość stypendium dla tej osoby, która ten staż wygrała, więc to jest bardzo dobra informacja, ponieważ ten staż będzie już miał trochę wyższą wartość. Firma zdecydowała się na 5,5 tysiąca, to jest jedna z tych informacji i ten staż dostała pani Magdalena Labuda. Pani Magdaleno, serdeczne gratulacje. Serdeczne gratulacje. Jeszcze jedna informacja od firmy Polpharma. Szanowni magistranci, już zaraz nasi absolwenci, firma Polpharma jeszcze się będzie kontaktowała z kilkoma z Państwa, ponieważ ma jeszcze jedno miejsce na staż i przeprowadzi dodatkową rekrutację, więc my przekażemy Wasze adresy mailowe, więc sprawdzajcie skrzynkę pocztową. Jeżeli pojawi się tam informacja, e-mail i zaproszenie od firmy Polpharma, to zachęcamy do tego, żeby się na tę dodatkową rekrutację zgłosić, ponieważ jest jeszcze jedno dodatkowe miejsce, które firma zdecydowała się przekazać dla naszego absolwenta na staż, więc to jest, myślę, bardzo dobra dla Państwa wiadomość. No i jeszcze...
I to nie jest fake news.
I to nie jest fake news. Więc szanowni Państwo, jeszcze raz serdecznie gratuluję, jeszcze raz bardzo Wam dziękuję. Przede wszystkim na koniec należą się ogromne podziękowania dla naszych studentów, organizatorów, dla samorządu. To jest ogromna praca. Tak, tam w reżyserce też. Bez Państwa ta konferencja by się nie udała. To jest ogromne Państwa zaangażowanie. To nie jest praca dzisiaj, wczoraj, ani 5 dni wcześniej. To jest praca przez kilka miesięcy, przygotowanie, strona, rekrutacja, wszystkie te informacje, które do Państwa spływają, to oni to robią tak naprawdę z własnej inicjatywy, więc za to Państwu bardzo serdecznie gratulujemy, dziękujemy. Gratulujemy też, bo niedługo Wy będziecie tutaj za jakiś czas, ale wtedy mam nadzieję, że się znajdą inni godni następcy, więc jeszcze raz bardzo serdecznie Wam za to dziękuję. Do zobaczenia na egzaminach i może wspólne zdjęcie jeszcze zrobimy. Myślę, że to będzie fajne na schodach. Przed wydziałem proponują, to możemy przed wydział? No to przed wydziałem. To zapraszam jeszcze przed wydział. Dziwnie się do tego mówi. Dziwnie się do tego mówi, nie? A, tu jeszcze dziewczyny.